All right, this is lecture number 2 of gene expression. Last time we talked about, we connected the dots, right? We got Garrett, the wonderkin of the early 1900s, where he's the one that noted, he made the observation that metabolic disorders were inherited in a Mendelian manner. So that was that key link there, and he called these inborn errors of metabolism. But between 1900 and 1940, not much was done in terms of understanding the mechanisms of a gene, right? And now, to be fair, right, we were still arguing about what genes were even made out of, right? And it's complicated. The paradigm of molecular biology is very complicated. The idea of DNA to RNA to protein, they barely understood any of it, right? So you got to cut them a little slack here. By the 1940s, though, we got Beadle and Tatum using the haploid and Rossberg crosser to come up with their one gene, one enzyme model. And that we went through that experimental protocol in detail. So you guys should definitely try to, if you don't already, make sure you understand Beadle and Tatum through and through. Ask me questions if you don't get it, and I'll give you the missing pieces of the puzzle in your head. We then go on to understanding hemoglobin, and understand that hemoglobin isn't one thing, it's actually 4 things, right? 2 different chains, alpha globulin and beta globulin. And they're the ones that finally realized that a gene encoded the information to make just a polypeptide, a chain of amino acids. Okay, and so the modern version of a gene, right? There's no, and really, I think in retrospect, the story should definitely have another piece here that connects the dots with RNA, right? We don't tell the story of RNA because I told you this a couple of times this semester. RNA is still something that we're coming to terms with. There's a lot to it. And we're just scratching the surface in this class on it. So, okay. So I'm going to skip all the Beadle and Tatum stuff. Here's hemoglobin. Okay, so this is where we're going to start today. I'm going to explain to you just very briefly what's going on in sickle cell disease. On the left-hand side there, this molecule, this is the beta globin. Is it globin or globulin? These 2 words are interchangeable in my head. Hemoglobin, alpha, beta. Is it globulin or globin? I always do this. I just call them alpha chains. Alpha globin. Yeah, it is globin. Okay, I was right. So you got 2 chains, 2 polypeptides folded up into 3D, and you got this, here. Remember this guy. So here, this is a relatively accurate, right, model of alpha globin or beta globin. So the idea is there is a slot in both of them, and within that slot sits a heme group, which is basically a carbon scaffold, and that heme group has in the core of it an iron ion, Fe, and oxygen, molecular oxygen, binds to it. Right, and so that's basically, think of it like a little docking port for a spaceship that goes in there. That's oxygen, in and out, in and out. The thing is, though, is every time oxygen goes in there, okay, if you look at the slide now, you'll see oxygen right here coming in and out. Okay, when oxygen comes in there, you can see it just nestles in there with the iron. Notice that the amino acids in the area around it are being pulled towards it a little bit. So the whole molecule ends up changing shape just a little bit. You see that, right? And if you look very closely right here, you see that little thing sticking out when only in, or I should say, it's the purple one right here. When oxygen is not bound to hemoglobin. So you see oxy, deoxy, oxy, deoxy, oxy, deoxy. When you pay attention to deoxy and look at the little purple nub sticking out. Okay, so in the absence of oxygen, hemoglobin has this amino acid jut out into space. And in sickle cell disease, the mutation over here changes what is a hydrophilic side chain, so that would normally be glutamic acid or glutamate, to a hydrophobic one. You see all those carbons, they're just, that's totally nonpolar, like oil and water. So the idea is when hemoglobin doesn't have oxygen bound to it, this becomes a hydrophobic area. And so what that does is it causes protein aggregation by trying to hide the hydrophobic residue from the water. And so you can see here in the middle, what happens, right? You could see that the hemoglobin forms basically these double filaments to mask that valine. And so you get these filaments. It's not supposed to be a filament. It's supposed to be a globular soluble protein. And instead you have these little hairs, basically, that causes the red blood cell to start twisting and contorting into that sickle shape, which renders it more likely to clog into arteries or capillary beds. And it will also vastly reduce its lifespan. And it's all of that from a single base change, from an A to a U. And as we'll learn at the end of all this, that's called, so that kind of mutation where you change one residue to another, one nucleotide to another, is called a point mutation. Okay, and there's different kinds of point mutations, and that right there is called a missense mutation. It's like misspeaking. It's saying the wrong word. You meant to say big, but you said small instead. Okay, and that's a radical missense mutation because we're changing its biochemistry, as opposed to a conservative missense mutation where the chemistry is still there. It's just a slightly different amino acid. So in other words, it would be like changing glutamate to aspartate, which is another acidic amino acid. Well, we'll cover that more in detail later. All right, we went over this the last time, the idea of the central dogma. We went through this slide here, transcription and translation. All right, we talked about the differences between prokaryotes and eukaryotes, how in prokaryotes it's only happening in the cytoplasm because that's all there is. And so they're happening simultaneously. And I taught you, does anybody, what's the word I used here? What's a special kind of transcript that only prokaryotes have? Begins with a P. Anybody? No, don't remember. Poly? Still no? Or are you guys talking and my hearing's not working? Cystronic. What's this mean? Polycystronic. All right, here we go. This is a big long piece of mRNA from prokaryotes. Over here is some open reading frame with codons. And over here, there's another one. Then over here, there's another one. Okay, this is unique to prokaryotes. Eukaryotes, one RNA gives us one polypeptide. In prokaryotes, we can, not always, but we can have multiple polypeptides. So you can very rapidly spit out a whole bunch of proteins from a single gene, or I should say a single region of the genome. Okay. We talked about the different RNAs: messenger, transfer, ribosomal, structural, functional. Okay, RNA polymerase 2 makes messenger RNA. RNA polymerase 1 makes ribosomal RNA, and RNA polymerase 3 makes transfer RNA. 3 different types of genes. The only ones we're going to worry about for our discussions is RNA polymerase 2. Just understand, all RNA is transcribed from a DNA, and there are genes. Okay, here's another way of saying it. Every RNA comes from a gene. You can trace every RNA back to a genetic source 100% of the time. Some viruses, their genome is literally RNA already. Okay. All right, so now we got to this in the afternoon class, but we didn't get to this last time. So I'm going to teach you how the language works now. Okay, this is the language of life. This is universal. Every cell on Earth speaks this language, which is mind-blowing if you think about it. Despite the fact that life is diverse, we have one language that unifies us. So much so that you can take a human gene and put it into a bacterium, and that bacterium will know exactly what it says and it will produce a protein. Okay, so here's how it works. The genetic code, okay, so let me step back for a second and say this. What I'm about to teach you isn't the only grammar of the language of nucleic acids, okay? There's two kinds of sequences in DNA. Right, the one we're going to talk about here is called a coding sequence or codon. Okay, a codon is a 3-letter word. Okay, remember we have 4 letters, A, T, C, and G, right, with the U replacing T in RNA. So a codon is 3 of those 4, and they could be the same. So you can have AAA or TTT or CCC. So how many codons do we have in all? How many codons does a language have? If we have 3-letter words and we have 4 letters in the alphabet? Good, 64, right? And the reason that way you get that is there's 4 possibilities, 4 possibilities, 4 possibilities. Remember in genetics, we had the rule of multiplication, right? Just pick, you can just think of the randomness there. Okay, so we got 64 codons. Remember that of the amino acids, there's 20 of them, right? So this language has to be rich enough to be able to describe all 20 of those amino acids, as well as tell us, hey, start making it, and then, okay, stop making it. So we need start signals, stop signals, and then amino acid signals. So within that 64 codons, all that's there. There's only 2 codons I want you to memorize. The first is the start codon, that's AUG. That means start translation. Now, I'm going to intentionally write the word translation there. Here's why. Some people will make a mistake and think that that's starting transcription. No. Yes, we start transcription there, but that's not what that is. I'll explain how transcription starts later. And then we have what I call the caveman codon, UGA, UGA. That means stop. Now, to be fair, there are 2 other stop codons, but I don't expect you to memorize all 4 of these, just 2, UGA and OG. Okay, now you can speak a little DNA, and you can also speak a little caveman. UGA means stop. Okay. All the others, except the other 2 we're not going to worry about. All the others code for some of the 20 amino acids. Now, some amino acids, I would say most maybe even all amino acids, it's hard to have multiple codons. Okay, so they might have 3 or 4 codons meaning the same thing, like say leucine. All right. When we have this situation where you have multiple codons, meaning 1 amino acid, the first 2 letters, these first 2 letters are always the same. And then third one is a play. It could 1 of 4 usually. So this is called the wobble effect. One of the purposes of the wobble effect is to guard against changes in the polypeptide chain that result from mutations. Okay, so think about it. If you have a random sequence of 3 letters and you change the last one, so you introduce a mutation to that, it doesn't change the polypeptide. It doesn't change the amino acid rate. So these are called silent mutations. That's another one of those point mutations. All right, so far you've learned 2 kinds, the silent mutation and the missense mutation. Silent mutations are the heartbeat of evolution. We can actually estimate the divergence of genetic sequences based on the accumulation of silent mutations within the same gene. So for example, if you compare our cytochrome C gene with a dog's and a monkey's and a starfish and a sea urchin and a reptile and a fish and a bacterium, you put them all in a row and just count the number of changes, it falls into the exact same row as if you were looking at a geological time record of fossils, down to within the precision of percent error of whatever your technique is. In other words, it all lines up. Further confirmation of evolution theory. Okay, so that's a wobble effect. Now, so this is the way it works. Here's the concept. We're going to take that piece of RNA, and that RNA is going to have some kind of code on it, right? The linker between it all is the transfer RNA, the tRNA. You can cartoon the tRNA like this. It's just the cartoony version of it. Okay, the key here is the TR. So this is, here, I'll tell you what, I'll color code this. So here, I don't like that. There we go. There's two aspects to the transfer RNA. The first is what's called the anticodon. The anticodon is antisense or complementary to the codon in the mRNA. And so these form hydrogen bonds. It's sticky. Okay, so that's called sequence-specific binding. That's sequence-specific recognition. Okay, there's a handshake there. And then over here, I'll use a different color here. Over here is the amino acid. Okay, which amino acid? Well, it depends. It depends on whatever this is. And right there, that's where the translation occurs, okay, in the languages. So in this case, UAC, well, really, that's the anticodon. So let's say the codon AUG, if you looked it up in a reference table, would tell you that that's the amino acid methionine. All right, so know that. So just know that one. Methionine is the first amino acid in the sequence of every protein ever made, ever, forever. Sometimes it's cut off later, but it's always there to start with. So right here, that's methionine. Methionine has the single letter abbreviation of M. It has the 3-letter abbreviation of MET. All amino acids have a 3-letter code and a 1-letter code. That's an easy one. Okay, so do you see that? The tRNA is the secret sauce. The tRNA is what decides what amino acid comes in with that anticodon. All right, so how did it get there, right? How did this amino acid get attached? So there's an enzyme. Of course, there's always an enzyme, right? So there's a special enzyme called, before I tell you what that is. Okay, this amino acid here, do you see how it's attached to an organic molecule, right? You guys remember functional groups? Remember how functional groups are called functional groups only if they're attached to another molecule, right? Like the hydroxide ion, OH is floating around, but if it attaches to an organic molecule, it's now the hydroxyl group, right? In the same way, an amino acid attached to another molecule could be construed as a functional group. So this here is called an aminoacyl functional group, technically. So this is what we're going to call this molecule itself. This is called an aminoacyl tRNA. Think of it as a charged tRNA. In other words, it's a loaded gun. It's got its amino acid. It's ready to be used. And so the enzyme that does this is literally, I'm just going to scroll over here so I can just add the next word, transferase. You're just making that up. You can't just put ace at the end of a word and make it an enzyme. No, that's actually what they do. The garbage truck is a pickup ace. It transfers stuff. It's a transferase. Okay, so this is a mouthful now. Aminoacyl tRNA transferase. Actually, technically, it's a synthetase. I'm sorry, I messed that up. Synthetase. Synthetase. It's making, it's not transferring, although it's semantics at that point. Really, it could have been named either one. It just depends on who names these things, but those are called synthetases. Sometimes you'll see other enzymes that are called synthases. There's no distinction, to my knowledge, between synthase and synthetase. It's just the person's preference who named it. If you discover something, you get to name it, right? So remember ATP synthase? Remember the enzyme that makes ATP? That's a synthase. This is amino acyl tRNA synthetase. Maybe they just wanted to put as many syllables in there as possible because it's already a big word. Who knows? All right, so here's the second reason why the wobble effect works. And in my opinion, this is probably the more important one. Think about the fact that cells need to be as energetically efficient as possible. Okay, actually, that's an important principle here. I want you to write this down because this is going to come up a couple of times today. Cells strive to be as energetically efficient as possible. If they can cut corners, they're going to cut corners. All right, so here's the insight. One amino acyl tRNA synthetase can charge all of the different tRNAs that have let me come up with a more systemized way here. I'm thinking of the wobble. So we have amino acid or nucleotides 1 and 2, and then X, whatever X is, right? Doesn't matter. So, and those would be different letters. Let's do this. So let's do X. Let's do this W, X, Y, and Z. There's their ATCG, right? So there's in theory, there's 4 different tRNAs that have the first 2 letters that are the same, then a different last letter, right? So a single tRNA synthetase can take care of all of those. Okay, so we don't need 64 enzymes, which are expensive to make. Now we just need maybe 20. Does that make sense? Raise your hand if that made sense. Okay, good. So it's an efficiency thing. So it guards against mutations, and it makes the cell not have to make so many proteins that it might need. Okay, so this leads to other interesting issues, such as the codons that our cells use, right? So think of these as codons, you know, A, B, C, and D, right? So each of those, I'm just making this up. Let's say that each of these codes for the amino acid leucine, hypothetically. In our cells, and let's say all mammals, let's say mammals tend to use these 3 codons for that amino acid, but bacteria tend to use this one. You see that? So codon usage isn't equal. The dialects between different species of different organisms has to do with the selection of codons they use. And in fact, some bacteria, or E. coli in particular, the lab strain of E. coli, and the reason I'm telling you this story is because I had to deal with this in lab, and it set me back about a year of work because it's crazy, because it's such a unique thing. I told this to Steve Gosshorn a couple months ago, and he's like, I've never heard of this. Nobody has unless they have to deal with it. It's called rare codon usage. Okay, I was sticking my protein, my gene, into bacteria to make it, and it was always making a small version of it. It made no sense. You look at the normal version, it was here. You look at the version I was making, it was here. It was a mystery. And finally, I was scratching my head for weeks and weeks and weeks. Tried all these different ways of purifying the protein. One day I was downstairs visiting the biophysics lab, the ones that do X-ray crystallography, and I was telling them my problem. And she immediately goes, the senior lab tech's like, have you tried using the Rosetta strain of bacteria? Anybody know what the Rosetta Stone is? Anybody remember what the Rosetta Stone is? What's the Rosetta Stone, April?
It was pretty much like how we could translate hieroglyphics from the Egyptians.
Good. Yeah. So there was this king back in ancient Egypt that wrote some royal decrees in 3 languages because his people, he had such a big empire. There were 3 languages. There was Aramaic, there was Greek, and there was Egyptian. And so the discovery of this tablet allowed us to say, oh, that's an E in Greek, that's an E in Aramaic. And so that must be what the hieroglyphic means, which means we can go into the tombs and we can decipher their artwork. So the Rosetta Stone was a key to understanding translation. So I call the chart, actually this chart here that we're going to look at in a second, let me get to it. I call this the Rosetta Stone of molecular biology. This lets us do all the translation. So anyway, the bacteria that I was using the normal E. coli strain to make the proteins didn't use one of the human codons. So I was putting a human gene into bacteria. The bacteria had no idea what to do with this codon, and so it just stopped there. It would just be like, I have no idea what to do here. Error 54. You know, it was like, they're not compute. The Rosetta strain of bacteria is an artificially engineered version of it that has all of the tRNA synthetases of humans. And so she gave me some of hers. She had some because she ran into this problem a couple of years ago. And I did an experiment side by side with my bacteria and the Rosetta bacteria. And when I ran the gels, here was my old one down here. Okay, so here the control would be this big. Here's the human version and the human cells. Here's my bacterial and E. coli. And then here's the one from Rosetta. It was so satisfying to see that after about 2 years of, maybe not 2 years, it might have been like a year. Maybe I'm exaggerating it now. Okay, so the idea here is this code is universal, but it's not always used in the same way. Okay, so now that we're here, you can see, right? Here are the 4 codons for leucine. You got CUU, CUC, CUA, CUG. And see you later. Sorry. There's your start codon. Right? There are your 3 stop codons, and there's UGA. So UAA and UAG are the other 2, but you don't have to memorize those. All right. You'll also notice that some amino acids, see if I can, I'm terrible at this, but where are they? Really? Can anybody else see them? There's 2 other serines. I can never find them. There's threonine. There they are. Oh my gosh, I'm so terrible at this. There we go. There's the 2. It's like, well, I was terrible. Where's Waldo too? Okay, so some amino acids are overrepresented in the chart, right? So serine has the functional group that has a hydroxyl group sticking out. This is what gets phosphorylated. Remember, I've taught you this before. Phosphorylation is one of the most important mechanisms in all of biology. Serine is serine's the one that gets phosphorylated the most commonly. Okay, threonine's the next one. Okay, and there's 2 others for threonine, which, wow, I can't find those now. If you can see the 2 extra threonines there somewhere. But the idea here is, actually, there might not be extra threonines, but there's 10 amino acids there, right? In other words, 10 codons out of 64 do one thing mostly. What does that tell you? It tells you that they're important. So the genetic code isn't random. This is not just a random collection of letters. This is a highly evolved language that popped out probably before the first cells came to be. Like, this was refined before we can see any record of it, which is kind of sad if you think about it, right? So the first cells on Earth probably used this language, which means before that first cell, there was a whole bunch of other languages, and this was the one that was the most efficient in one. Okay. All right, so some rules of the language. Let's go back here. So the way this was hammered out, the way we discovered this, there was this group of researchers who used basically precursor red blood cells from rabbits called reticulocytes. A reticulocyte is an immature red blood cell. It's a protein factory. It's got lots of rough ER. That's why I call it a reticulocyte, reticulum, endoplasmic reticulum. It's got lots of rough ER, lots of ribosomes, lots of amino acids, lots of tRNAs. All you have to do is lyse the cells and then add your own artificial RNA. And it will make your amino acids. And so what they did, the very first experiment they ever did was just a poly A. They just put a whole bunch of A's in there. And what popped out of that was a bunch of phenylalanines. And you can imagine how exciting that must have been to ask a question or to make a statement, A, and then get an echo back, phenylalanine, 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 right? And then how about U, or CCC or GGG, right? So they basically artificially translated a bunch of artificial RNAs, and they got sequences out of it to the point where they got really good at it, where they could do this. And let's look at this sequence for a second so we can understand this. That's just a repeat of UA. All right, so but I want you to see this. Okay, so that codon is UAU, right? But what if so that's if I started reading at position 1. What if I started reading it instead of position 2? Then I have the codon AUA, right? Do you see that? And what if I start reading it in position 3? Then I have UAU again. Okay, so this actually isn't the best this isn't the best example of giving you all the reading frames. So let me just add a third letter here. Let's say C. All right, so let's say UAC, 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 UAC. Okay, so we start reading in the third one. So we start reading here, right? We get UAC. We start reading the second one, we get ACU. So let's ACU. And if we start reading the third one, let me clean this up. There we go. We get CUA. But what if I start reading the fourth one? Well, we're back to the first one again, right? UA, let me change that there. Now it's all there. That's all consistent now. So you see how there are 3 different start points in theory. And so therefore, you're going to get 3 completely different codons, right? What's the second codon in this version here? That would be UAC, right? UAC. So that's a repeat. How about this one? ACU, ACU, AC. Okay, so they're all repeats, right? That makes sense. And then CUA, CUA, CUA. So we call these different start points reading frames. So there are 3 reading frames in the language of nucleic acid. There are 3 coding reading frames. And so a reading frame that encodes a specific a codon sequence for a polypeptide chain, we call that an open reading frame. You can abbreviate that ORF. Okay, so associate ORFs and codons. That means you're going to make a protein. There's a UAG at the front of it, and there's a UGA at the end of it. Okay. Any questions so far? All right. So you can imagine if you start reading in the wrong place, nothing's going to make sense. Take the simple English sentence, the red dog ate the bug. And then start reading in the second reading frame. Her ed aga tet heb ug. I have no idea what that means. That doesn't mean anything in English, right? I mean, none of these are words except ug and her. Well, her is a word and ug. I mean, I don't know if that's spelled. I think we usually put an H at the end of that. Ug. Anyway, the point is that it's different and you can't predict it. Okay. So another mutation is if you insert into a genetic sequence 1 or 2 letters, right, and you shift everything, that would be called a frame shift mutation. A frame shift mutation changes the reading frame. And the impact of a frame shift mutation is you now have a completely new random stretch of amino acids that could not be predicted. And so you can finally see at the core of it all, the power of a genetic mutation in terms of forging brand new things, right? I mean, it's random, but if you do that 1 trillion times, you're going to make DNA polymerase. right? You're going to make brand new enzymes. That's the power of the frameshift mutation. Now, normally, frameshift mutations are terrible because you're introducing a whole bunch of randomness there. And so imagine a photocopier that started going haywire, or a printing press that started going haywire. What do you do? What do you try to do immediately? What would you do in that kind of situation? If you're working with a machine and it just starts going crazy, what are you going to want to do?
Unplug it.
Right. Just shut it down. That's why there's 3 stop codons. Okay. If you had a 64-sided die and you were rolling it randomly because of a frame shift, you have a 3 out of 64% chance to stop the madness before things get too out of hand. Okay. There's a reason for all this stuff. Okay. All right, yeah. So the first RNA was a poly U. All right. So I want you to look at that chart and I want you to see how to read that. You're going to sometimes see this chart in different ways depending on where you go in life and what you're looking at. I don't like this one at all. I prefer the rectangle one, but this is another one. So you start by reading in the first ring, and then you go to the second ring. So here's the first ring, here's the second ring, and here's the third ring, right? So you look around the ring and you'd be like, okay, you, right? So you, there it is. There's you, phenylalanine. Okay, so here, I'll give you a random, let me just think of a random one here. How about CCA? Go ahead, whoever, the first person to find it, shout it out. Just give me the 3-letter abbreviation.
Pro.
What'd you say, Julia?
Pro.
Just spell it out.
PRO.
TRO. There's no. Wait, let me zoom in a little bit. Maybe you can't see it because it's all fuzzy. All right. So what did I say? CCA? Yeah, CCA. All right. So ready here. Here's CCA. So that's TR. That's THR. It's pixely on my end. So on yours, probably that I can see that looking like an O. That's threonine, by the way. Okay. So that's how you do. That's the skill you're going to do. And your homework has a couple of questions where you'll get to look this stuff up. So these were deciphered in the mid-60s, right? Just think about that for a second. Oh, go ahead, Juliet. Do you want to say something?
Wouldn't you start at the C in the middle?
CCA.
Yeah, because there's met
Actually,
and
that,
says
you know
start
what?
underneath.
That actually makes more sense. And yeah, actually, you see the 5 to 3 prime. And we read things 5 to 3. So good job, Julia. Yeah, so you were right. So did you say PRO?
Yeah.
Yep. Okay. That's proline. And yeah, that makes a lot more sense. That's why I was like, when I was I did you, right? So I was like, where's you? Where's you? So well, I guess because it was all U's, it didn't matter which way I was reading it. Right. But CAA, yeah. So okay, you start in the middle, and that makes
I
it a
thought you
lot
said
easier.
CCA.
Well, the very first one, this one, when I did this talking out loud to you, I was looking around and I did this at first. I just found the first U I could find out here. And then I found a second U and then the third U. And then I realized that it was over here. So I did it ass backwards. It was terrible the way I did it. And the thing is, I was starting from the outside going in because U doesn't make sense. But the CAA, that does, right? AAC would be a completely different codon. Right. So as Julia pointed out, if you start in the middle and you branch out, it's also easier to read that way. Yeah. All right. And I got a chat message here. Oh, pro. I'll keep that up now. All right. So the way I like reading it is this one over here. So the way you use this table, and this is what I mean. I had this printed out and stitched to my wall when I was in grad school. So first base, second base going this way, third base going this way, right? So you'd say U. That's how your eye looks. Your eye looks here, then it scans at the top, then it scans at the side, right? And you triangulate. Okay, this also lets you see the wobble effect as well. You can see, right, almost all of them have that first 2 amino acids being the same.
Question.
What did you say, April?
So you can use both of those charts to read.
Say that again.
You can use both of those charts that you just showed us to read the codon then?
This one here.
Yeah.
So what's your question?
So are they like, if you give us on like the test, like are those the same?
Oh yeah, yes.
Yeah.
Yeah, you would not have to memorize this.
Okay.
Yeah, nobody memorizes this. I'm sure there's somebody that's memorized it. Those are the same people that memorize pi to the 30th decimal point or whatever. All right, let's move on. Okay, so here are the formal rules of the road here. The language is degenerate. That's the phrase that also means redundancy. So a degenerate code is a code that has multiple words that mean the same thing. That's the whole wobble effect. It's unambiguous, meaning every word always means the same thing. You can't have a word that means 2 things. The exception there being the start codon, but that's not meaning 2 amino acids. All right, there's start and stop signals. There's 1 start codon, there's 3 stop codons, right? You got uga, uag, and ua. You don't have to memorize any of those, just uga. And then the code is universal. And then there's a little footnote with some exceptions. So in other words, the words mean the same thing for every single organism, which supports the laws of evolution, showing that we all come from a common ancestor. Our cells speak exactly the same complex language. And that should be humbling. And there's no punctuation. It's just one big long run-on sentence. Now, that said, codons are just one type of word, okay? There are many, many other words, okay? In general, the other words are sequences that proteins bind to, okay? And so we'll see a whole bunch of that soon. And so, right, so the codons sit right next to each other. There's no gaps in them. And if you know about introns and exons, that's not what we're talking about. We're talking about the actual coding sequence. Okay. This, I think, is from your book. I think this is another, and it's just a different way of laying it out. Right, so you can see that this, if you just step back and look at this, you can see more easily the multiple codons that correspond to a single amino acid, right? And so here, look at cysteine. See how cysteines only got 2 amino acids. Okay. Where, you know, arginine has 6, serine has 6, histidine's got 2, glutamine's got 2. So there's a reason for all this. Then you'll actually dig into that when you get to molecular biology someday. Here are the 20 amino acids as a reminder. Okay, see the different color codings? You can see that there are different families of amino acids. Different people divvy this up in different ways depending on who's in charge. So you could see here, these are acidic, these are alkaline, these are polar, these are non-polar. That's the easiest way. So here, there's non-polar, polar, or charged, acidic, and alkaline. There are many ways to split this up. This is the simplest, and you don't have to memorize this. Just understand all the chemistry we've talked about all semester is represented within the 20 amino acids. Okay, so that's all I'll say about that. Here's a schematic that represents the central dogma. You got your DNA sequence here. Okay, let's zoom in a little bit here. You got your DNA sequence here, AAA, CCG, GCA, AAA. You can see that the antisense of that, UUU, GGC, CGU, UUU, right? So that's the RNA. So they're complementary. So this would be the template strand of the DNA. And the other strand is not even here. That's the non-template strand, right? And then the RNA itself, you can see these 3-letter words, these codons correspond to a particular amino acid. And you can see why none of this work was done from 1900 to 1940. This is just insanely complicated, right? And that over here, this is the ribosome that translates it all. Okay. There it is, bigger. There's transcription and translation. You can see now tryptophan, phenylalanine, glycine, serine. So you could see UGG stands for tryptophan, UUU stands for phenylalanine, etc. All right, and now you can see here's the double-stranded DNA. All right, so here, this is what we'll do. Let's go to the whiteboard here. Okay, ready? We got our DNA. Right, we got our 5 and 3 and our 3 and 5. We have our RNA. And we'll call that our messenger RNA. And then we have our polypeptide. Notice I'm not calling that a protein. It's not folded up. What I want you to see here is the relationship between the complementary or identical sequences. So here's what we're going to say. Everything is relative to the RNA. So we're going to say this is the sequence itself we're looking at. All right. If we called this strand here, if that strand here, let's write it on this side here. This strand here we'll call the template strand. This strand we're going to say this is a non-template strand. This is the transcript. And this is the polypeptide. I'm just going to abbreviate that PP. Okay, so what I want you to understand is what's the relationship between, say, the transcript and the template strand, or the transcript and the non-template strand. All right, so let's introduce some codons here just for the sake of looking at things. Let's say this is AUG, GUU, ACC. All right, so let's work this out now. So the template strand, right, we can all agree the template strand has to be complementary to that, right? That just makes it, because it's a template. The template's what we're going to read. So that's got to be complementary. So we're going to call this a sense strand. And this is going to be antisense. I'm sorry. This is the antisense strand. Antisense means complementary. Okay, when you see the word antisense, I want you to think it's going to stick to something, like Velcro. Okay, so you would say that the transcript is sense to the non-template strand. In other words, the non-template strand would say exactly the same thing, except with the T's instead of the U's. Whereas the template strand would be antisense or complementary. So the word antisense, you can also say complementary. So the codon anticodon is sense antisense. So you can understand coronavirus now. Ready? The coronavirus that we're dealing with in the pandemic, its genome is a positive RNA, which means it is essentially this. The coronavirus genome acts basically like a messenger RNA when it gets into our cells. Okay, there's negative stranded RNA viruses. And so that would have to first be transcribed into a sense strand to be made into proteins. Okay, raise your hand if you're still following all this. Okay, cool. All right, so that's it. Now I'm gonna go into the actual mechanisms here. Yeah, this is amazing, right? All the different, these are just samplings of life, no matter how. So there's a tardigrade, a tardigrade to tigers, to sea squirts, to strawberries, and roses, and diatoms, and cuttlefish. They all speak the same genetic language, and they use the same 20 amino acids. And this terrifying spider down here. This is a petri dish that has been painted with different strains of bacteria that have all been artificially transformed to express various fluorescent proteins. These fluorescent proteins here, such as this is enhanced GFP, green fluorescent protein, citrine, okay, M turquoise cerulean. These are all, we find all these different kinds of proteins mostly in jellyfish. We can extract the genes from jellyfish. We can engineer them into plasmids. We can put the plasmids into bacteria, and now you can have some really bad art. Okay. Now, the next few slides are just for you to go over if this is really fascinating stuff to you and you want to dig into it a little bit. These are exceptions to the rules. This slide here says mitochondrial codon usage exceptions. For example, in animal mitochondria, AUA does not mean isoleucine, it means methionine. Okay, but I'm not going to read all this. And then you got nuclear gene exceptions, which are very rare, right? So for example, some protists, some ciliates use 1, 2, or 3 of those stop codons to encode for different amino acids instead. Okay. And then we have, this is probably the neatest one. There's different amino acids, non-standard amino acids. One we use in our bodies is called selenocysteine. Well, we use both of these actually. There's pyrolysine and selenocysteine. And we still don't fully understand how this stuff works. So you can see here in the pyrolysine, actually, we don't use pyrolysine, we use selenocysteine. Pyrolysine is only in prokaryotes. And UAG is used for interchangeably as stop or pyrolysine, and we have no idea how this discrimination occurs. It's a mystery. Okay, so this is the cool exceptional stuff. Okay, here we are. This is the big picture now. This is where we're going to dig into mechanisms here. This is the song of gene expression, and there's 3 parts to it. There's transcription, there's post-transcriptional RNA processing, which you really can just abbreviate this when you talk fast, RNA processing or mRNA processing, and then translation. All right, now notice that steps 1 and 3 here, transcription and translation, each have the same 3 phases: initiation, elongation, termination. Initiation, start doing it. Elongation, keep doing it. Termination, stop doing it. So there's nothing surprising here. Each one of these phases has their own rules and players, and we'll talk about those. Okay, post-transcriptional processing, this is basically surgically preparing the mRNA to go out of the nucleus, right? So this stuff happens, with the exception of this one, this all happens in prokaryotes and eukaryotes, right? Obviously, there's no nucleus to export out from in prokaryotes, okay? And bacteria don't splice. So here, let's do this. Not in bacteria. Notice I said bacteria and I didn't say prokaryote because archaeans actually do that. Not in prokaryotes. Neither of them have the nucleus. This is universal. Okay. So I'll teach you as we do all this, I'll give you the exceptions between the prokaryotes and eukaryotes too. All right, so I'm going to go through this once and I'm just going to do a straight run through it. If it doesn't make perfect sense, go back and watch it again. Okay. The first thing that happens. So again, let's look at big picture here. Let's keep this organized from the start here. Okay, ready? We're learning gene expression. I'll tell you what, I'll give you the cartoony simple version first, and then we'll flush it out. So I'll give it to you a couple times. Okay. You guys want me to do it this way? Do you want me to use slides? Raise your hand if you want whiteboard. Raise your hand if you want slides. Okay, slides. So here we go. Initiation, elongation, termination. All right, during initiation of transcription, there is a series of proteins that swarm the DNA into a region called the promoter. We call it the promoter region. As its name suggests, it promotes transcription. Okay, so these proteins bind to the promoter, and they will then open up the DNA, and then they will start reading along it and produce. So initiation is all about the binding and the denaturing, the melting of the breaking apart of the strands. Okay, and then so think of it like a train on tracks. Its engine just starts getting going here, and then it starts going. Here's your elongation. During elongation, you can see an RNA transcript is emerging from the system. Okay, and again, it's always 3 to 5 prime, from the 3 prime to the 5 prime, right? Because we're reading it from 5 to 3. So you're going to see the 5 prime end is the first to pop out, right? So the cartoony version of that would look like this. This right here is the 5 prime end, because we're adding this way. Right, there's the 3 prime extending. All right. And then termination occurs in completely different mechanisms in prokaryotes and eukaryotes. So in eukaryotes, you can think of it as the polymerase just falls off the DNA. Eventually it gets bored and goes away. It's a messy end. In prokaryotes, there's different mechanisms. You don't have to worry about any of the termination stuff. Just understand it stops. Okay. So here is a gene, the generic version of a gene. Genes all have this upstream. So you see upstream, downstream, that's how we talk. All right. So when we say upstream, we're going backwards. When we're going downstream, we're going forwards. We read downstream in English, you can think of. Okay. So the promoter is the regulatory region. Okay. And really, that's the core of the regulatory region. There's other regulators. Okay. So for example, some promoters will have sequences here, right here. This is the DNA. Let's say we go really far away over here. All right. So there are sequences of DNA really, really far away that can loop back, because it's 3-dimensional. It's like a wet spaghetti noodle, right? It can loop back and come to the promoter. These are called regulatory sequences, regulatory sequence. These would be cis regulatory sequences. Cis and trans. Cis is close, trans is far. Okay. These are generally referred to as enhancer regions. So an enhancer is something that makes it even better. Okay. I generally don't talk about these. That's just wrapped up into the promoter. Okay, so promoters have binding sites for other proteins. And then, so already I'm going to delete all that now. There is a start site. There's an ATG right here. So when I say there's an ATG here, right? Let's understand. When I say ATG, that's the DNA sequence, right? That's actually the non-template strand, because when I say ATG, you recognize that as a start codon, AUG, ATG, right? But understand, oops, ATG, right? TAC, right? This is actually the double-stranded DNA, right? And so this would be the coding or the template strand. This is what the RNA is going to say. So when we talk about DNA sequences, we almost always, we always report them out as the non-template strand, because this non-template strand is the sense strand to the transcript. Okay. For example, if you're a researcher and you're looking at a DNA sequence and it's a coding region, you want to just be able to look at the codons and oh, that's a phenylalanine, that's a serine, whatever. You don't want to have to do the complementary translation first. Okay. So there's our ATG start site. And then this whole thing right here, there's our open reading frame. Your book will call that a transcription unit. At the end here, okay, there's a sequence called, in eukaryotes, we're going to call this the polyadenylation signal. I'll tell you what that means in a second. And in prokaryotes, we have generally what's called the terminator region. Okay. And that's the gene. So the promoter is the DNA sequence that defines where the gene transcription begins. It determines whether or not the gene is on or off, right? It's regulatory in nature. And it dictates where, ready, RNA polymerase, RNA polymerase, RNAP 2, put the Roman numeral 2 there, RNAP2, RNA polymerase 2. The promoter dictates where it binds and it tells the polymerase which strand to start reading. Okay. And so here's your first, what's called the consensus site. Consensus site. And it drives me nuts your book doesn't use this phrase. A consensus site is just one of those sequence-specific binding sites for proteins that I told you about earlier. Okay. And so there's this thing called the TATA box, which is a name that makes people giggle usually, but it's just TATA. The concept of a consensus site is the word itself is not always exactly the same. Some TATA boxes might have another A here, or some of them might be TTA. I don't think that's common though. All right, but the idea is there's some play in it. Okay, so the TATA box has a protein that binds to it called the TATA binding protein, or TBP. TBP. And so TBP is your first introduction into the world of transcription factors. A transcription factor is a protein that regulates transcription. There's positive regulators and there's negative regulators. TBP is a positive regulator. Okay, now here's the other thing. And so everything I'm about to teach you here this is all eukaryotes, in particular mammals, but you can use this as a general concept for all of it. Every cell in your body needs a set of genes on all the time, right? I mean, we call these housekeeping genes. Think about, the genes necessary to make ATP and lipids and proteins, blah, blah, blah. So those genes that are always on, we call those constitutive genes. A constitutive gene is a gene that's always on. You would say it's constitutively expressed. Constitutively expressed genes have a TBP site or TATA box. Okay, so because this is not specific to a specific set of genes, but kind of universal, right? So 50% of our genes, 50% of our promoters have a TATA box. We call this a basal transcription factor. Basal meaning like the basement, universal. Okay, so there's basal transcription factors and there are specific transcription factors. Okay. Raise your hand if that made sense. Okay, you guys kind of like absorbing this like a sponge right now, or are you getting overwhelmed? Give me some feedback here. You guys okay? Okay. All right. So there you go. So promoters contain specific words, specific sequences called consensus sites, which can recruit specific proteins called transcription factors. So here, what I'm about to teach you is this is the biggest difference between prokaryotes and eukaryotes in terms of how they work. Ready? So that's a P for promoter, just to make this easier. Okay, in prokaryotes versus eukaryotes. In prokaryotes, RNA polymerase can bind to DNA directly. There's just one RNA polymerase. There's not RNA polymerase 2, it's just RNA polymerase. It does all of it. Don't worry, Jordan. Glad you're back. Okay, in eukaryotes, RNA polymerase, all of them, RNA polymerase 1, 2, and 3, RNA polymerase 1, 2, and 3 cannot bind to DNA directly. Okay, so in other words, they need a protein that can bind to DNA to anchor them there. Okay, so you would say that it is recruited to the promoter. RNA polymerase is recruited to the promoter by transcription factors. And I abbreviate transcription factors TF by TFs. So does that make sense? Right? The way I see this is there's a swarm of protein, and I'm not going to teach you the complexity of this. There are a lot of transcription factors that start this in eukaryotes. They swarm the promoter like a bunch of ants carrying a banana. That's the idea of RNA polymerase, right? So we've all seen at one time or another in our lives, we go outside and we see a bunch of ants carrying something as a group, right? That's kind of how I see the protein swarming onto the promoter and then recruiting the RNA polymerase. All right. So transcription factors, right? These are proteins that regulate gene expression by binding to the promoter, and then they either recruit or block polymerase. This is what makes them plus or minus. Okay, so there's transcription activators and there's transcription repressors. And notice we call them repressors and not inhibitors. We repress gene expression. We don't inhibit gene expression. It's just the English of it. Okay, so I want to teach you two of them. These are the ones I worked on, which is why I'm teaching them to you, because these are the ones I know the best. An example of a gene activator is E2F. E2F is a positive regulator or an activator of genes in S phase. Right, so think about that for a second. E2Fs bind to promoters of genes that we need to be on when we go into S phase. Go back to the cell cycle. Remember, we go from G1 to S, right? The way we go from G1 to S is by turning genes on that go into S phase, and E2F is one of those players. Okay, and then RB, the retinoblastoma protein, PRB, is a repressor. It also happens to be the first tumor suppressor discovered. A tumor suppressor, as its name suggests, suppresses the growth of transformed cells. So it is a negative growth regulator. Its job is to bind to promoters of S phase genes and stay there. Okay, so here's the concept. This is active repression. That's a key concept to understand the way regulation works in. Okay, active repression, meaning you're keeping your hand. So imagine there's a, let me think. Okay, you know when you open the refrigerator and the light goes on, right? You shut the door and the light goes off. You open the door, the light goes on. Think about when you open the refrigerator, right, from a side view of it. If this is your refrigerator side view, there's a button in there, right? And when you push down on that button, right, when your finger is pushing that button, right, so this is on and this is now off, right? As long as you keep holding that, it's off. You are actively keeping that button off, right? There's a little spring in here. That's how it works, right? So when you let go of it, it pops back open again. Okay, so that's an example of active repression, which means if this protein suffers a loss of function mutation, now the genes that it turns off are accidentally going to be turned on. And that's what leads to carcinogenesis. That's how cancer works. Okay. All right. And I know I'm hitting you with a lot, but you can pause this and rewatch this later or whatever. So we got E2F activates S phase genes. We have PRB that inhibits it. Okay. There are always these tug of wars going on here. And so there's things that activate this, and there are things that inhibit that, and vice versa. And things start to get messy, and we're not going to go there. All right. Now the next layer of complexity. Eukaryotic genes and archaeans within their open reading frame contain two fundamental types of sequences. They're called introns and exons, or exons and introns. The exons are the important thing. Think of them as the expressed codons. So we want just the exons. We don't want the introns in our RNA. Okay, think of the intron, and different books use different phrases. I say they are interspersed codons. They're not codons, they're interspersed sequences. They're in between. So intron in between. And I think another way of doing it is region. That's where the ON comes from, region. Interspersed region. Expressed codons. You could say expressed region. It doesn't matter what they stand for. The point is that the exons contain the useful codons to make the protein and the introns don't. Okay. So I want you to imagine a gene sequence that looks like this. Here's exon 1. Actually, let's make it nice and short here. Here's exon 1. Here's intron 1. And here's exon 2. Okay, so exon 1, exon 2, intron 1. I'm just to illustrate this simply, I'm going to show you this. So let's say that we make the piece of RNA. Okay, so we do transcription. And so what we're looking at here now, we're going to call the primary transcript. Okay, so this is the piece of RNA that we've just made from transcription. RNA polymerase 2 made this. Over here is its 5' end. Over here is its 3' end. Okay. And so I'm going to skip ahead here and go into RNA processing and just give you the quick steps here. The first thing we're going to do is we need to protect this end from being enzymatically degraded. We need to protect the cap or the end. And so we're going to cap it. And the way we do that is by adding a special methylated guanine. Okay, so this is methyl, think of it as methyl G. I don't know, I guess you can say G subscript M. It's a methylated guanine. This is called the 5' cap. And it protects it from enzymes called RNA exonucleases. Think of them like Pac-Man. So that wants to eat the exonuclease from the outside. An endonuclease would cut it from within. We're not talking about those now. Okay, so we protect the 5' cap. Then over here at the 3' end, there's a signal in here that I said to you earlier is called the polyadenylation sequence. So there's an enzyme that binds to that. And that enzyme is poly-A polymerase. And the job of poly-A polymerase is to basically add a whole bunch of adenines here. Here, I'll make it neat first. So A. It knows one word. It's not a very good polymerase. It just knows one word, A. Okay, so we call this the 3' tail. Or sometimes we say the 3' poly-A tail. We say that the process is polyadenylation. Right? Okay, so now let's go back to these exons. So here's how you doodle a transcript. If you have multicolors, you can go like this. There. And we call this the mature transcript. Oh, actually, no, we don't. No, we don't. Don't start. Just stop writing that because we still have to get rid of the intron now. Okay, so this last thing we're going to do, so we capped it, we tailed it, and now we're going to splice it. And here's how the splicing works. There are sequences recognized here called boundary elements. And they are recognized by this big special enzyme. Well, really, I'll tell you what, let's make it simple. Let's just this whole thing gets bound. This is called the spliceosome. Splice within E, splice, and then O-zome. So it's spliceosome, but it splices it. The spliceosome, like the ribosome, is made out of protein and special RNAs we're not going to learn about. Okay, there are small nuclear RNAs called snRNAs, but we're not learning that in this class. Okay, but the idea there is those RNAs are in there and they recognize these boundary elements. We can recognize the intron-exon boundaries. And when they do, this is what the spliceosome does. It will bend that RNA such that it loops out that intron. Cap, tail. See that? So the spliceosome, let's not show the spliceosome. It loops it out, and then what it does is it cuts this, and it ends up releasing it. So the intron is cut out and released. And what that does is it results in the two exons, which are now pasted together. All right, so the spliceosome also ligates them together. And you now have your mature transcript. And this gets exported to the cytoplasm. Okay. All right, so I'm going to go back to the slides now. I'll show you all this. All right, so there's the open reading frame, ORF. This is the sequence that contains codons. Okay, your book calls these intragenetic regions as opposed to expressed regions. introns are small compared to exons. I'm sorry, exons are small compared to introns, despite what your book shows. That's why when I drew it, I went like this. Here's the exon, here's the intron, here's another exon. So in reality, introns are big, but for the interest of diagrams in books, they generally make them all the same. But there's a scaling issue there. And the reason is the introns kind of protect everything, right? Think of a random mutation occurring somewhere. Chances are it's going to hit somewhere in the middle of that intron. So it doesn't matter. Okay. Prokaryotes don't have introns. They're just one bit. So we don't even say they have exons. We just say they have an open reading frame. We don't use the terms introns and exons in prokaryotes. Okay. All right. So ready? Step one, initiation. You don't have to memorize this. This would be if we were taking molecular, if I was teaching you junior level molecular biology, you would have to know that. All right, you don't have to know that, but I'm going to show it to you so you can get an idea of how it works. Okay, so here's a promoter, and you can see here, if we zoom into it, TATAAA. Okay, so that's a Tata box. So that's just a little variation of it. All right, you see the little, it's hard to see this because it's pixelated, but that says +1. Okay. You see how the TATA says -30 and this says +1. So these numbers correspond to the distance from the start codon, AUG. Okay, so that serves as our anchor point. So we would say that the Tata box lies 30 bases downstream from the start site. Okay. And in fact, if I handed you the coronavirus genome and I asked you, hey, how many open reading frames do you think this thing has? You can read through it and you can look for TAA and you can look for AUG. And if they're 30 away from each other, maybe you could call that an open reading frame. That's the way things work. All right, there's TBP, Tata binding protein binding to the TAA site, right? But then there's this guy here, you see it says TF, and then those are, it looks like 2 I's, that's a Roman numeral 2 for RNA polymerase 2. So this is transcription factor for RNA polymerase 2, and this is transcription factor 2D, because that was the one that it was discovered fourth, I guess. So this is TF2D, right? So this is how you would tell the story. For this gene to turn on, Tata binding protein binds to the Tata box, recruiting TF2D. This complex in turn recruits TF2A and TF2B, which in turn recruit TF2E and TF2H. And once you have this called the pre-initiation complex, that would recruit the RNA polymerase 2. Okay, for us, for what I want you guys to understand is I just want you to understand the concept of the pre-initiation complex, pre-initiation complex, which is made out of transcription factors called TF2s. And I want you to see that as a swarm of insects on a piece of fruit. They just all swarm down there. All right. Once the pre-initiation complex forms, that recruits the RNA polymerase. And now we call it the initiation complex, the transcription initiation complex. All right, so pre-initiation, because we're not started yet. Right? The party doesn't get started until the polymerase is there. And then the polymerase denatures the strands, pulls them apart. And by the way, this pre-initiation complex and all that complexity there, that cluster decides which one of the 2 pieces of DNA is the template. I've had that question a couple of times throughout the semesters where I'll just teach it to you. Is people always ask, like, how does the RNA polymerase know which one to read? The pre-initiation complex tells it. So it depends on the transcription factors present. Hold on one second, guys.
as we cook it in the
40 seconds. Big deal. Jack just asked if he can make himself a hot dog in the microwave. Let's see how this goes. So expecting about 3 or 4 minutes, he's going to run in here and tell me something is going to. All right. Any questions?
So this is kind of like a stupid question, but like it's on a
No stupid questions at this point. No, it's not stupid.
It's on a strand of RNA, right?
Say again? Why can't I hear
This
you
like
well?
complex that you just talked about, it's on a strand of RNA, right?
No, that's a great question. No. So here's your double-stranded DNA. Everything we're talking about is on the DNA. Okay, so the promoter is this region of double-stranded DNA. Right, so the Tata box is actually right? It's actually double-stranded DNA. And so the TBP binds to the actual DNA. So here, so my little. Okay. And then let's use this. Okay, so here, look at me for a second. Here's double-stranded DNA. Here's my little 3D printed DNA. See this? Okay, so if you can imagine if this was flexible enough, I could kind of untwist this and make it look like this screen, right? So there's like a TAA, TATAA. So there's a protein that will literally bind to those sequences. Okay, and then another protein in turn. So, wow, that was fun. So this interaction here is sequence specific, right? Actually, this works really nice. I never did this before. Here. This is hard to do though. Now, okay, let's try this one more time. So here's TBP. Okay, it binds within the groove of the DNA in a sequence specific manner, right? And then TF2D would come along. TF2D doesn't bind to DNA, TF2D binds to TBP, right? And so you can imagine now the swarm that comes around the DNA that essentially culminates into something that would probably look more or less like this. Okay, so this is the pre-initiation complex. And now here's the prima donna RNA polymerase 2 that has to be, escorted along here. And so this thing can't bind to DNA directly. It has no idea, just floating around, but it can bind to all these proteins here like this. Boom. Okay, and so that tells it, go here, use this strand here, mount this apart, and then start moving along the DNA. Okay. So the only interactions with the DNA are with the transcription factors. Now in prokaryotes, promoter, polymerase, much simpler. Okay. So we need a different way of regulating that, and we'll teach you that in a second. Any other questions? So we have no RNA yet, Alexa. We have not made a single piece of RNA yet.
Okay, so it's the DNA strand that like there's the promoter with like the Tata box, and then the TPB comes to that, and then all of the TFs, the transcription factors, go to that, and then the RNA polymerase goes to that.
Exactly. You got it. Think of it like at the very end of it all, we put the crown on top. Right, and that's the RNA polymerase 2. Hold on a second. What? Oh, my prediction's coming true. What's up, Jay?
I think I opened the hot dogs the wrong way. I don't know how do I open? How am I supposed to open them?
Just do your best.
I got 3 of them at once.
Okay, put the others back in a plastic bag.
I've been trying. They're stalled together.
Get them. You got okay, guys. Give me 1 second. I gotta go help myself.
Sorry, Dad.
It's okay. It's okay. I'll be right back. box there. Whatever he's learning, that's good. Okay. So this is the level of detail I want you to know. Okay, there's a promoter, it binds the transcription factors. The transcription factors recruit the polymerase. Easy peasy. Okay. So that's initiation. This is how you turn a gene on. When we talk about genes being turned on in cancer or genes being turned off due to pharmaceuticals, okay, this is how it works. All right. I said all this to you already. There's nothing new here. Oh, I didn't say this. It doesn't need a primer. It is a primer, right? So it doesn't need to be primed. It's not like DNA polymerase, right? So remember, DNA polymerase needs an RNA primer. RNA polymerase doesn't. It just goes. Okay. So another question I usually get is, well, where are all the nucleotides coming from? There's this idea of the super round, the metabolic pool. Okay, so all of the things that are needed to make anything in the cell are just floating around. That's the way to think about it. Okay. All right, and so this whole thing that we just learned, this is true for all the other types of RNAs, the tRNAs, the rRNAs, the mRNAs, the small nuclear RNAs, the non-coding RNAs, the SNPs, the SNRPs, whatever. Okay. Okay. Elongation's easy. We just ticker tape along, as I say, we ticker tape along the template and we make a piece of RNA. We just go. Okay. Now, this can have errors, but we don't talk about errors. All right, because here's the key. And I want you to think of it this way. Here's a DNA. I want you to imagine a whole bunch of polymerases bound to that DNA. And each one of those polymerases is making a piece of RNA. So one gene can give rise to thousands or tens of thousands of transcripts. We would call this a highly expressed gene. Okay, so yeah, mutations can happen in there, but it's just one in a sea of others. Okay, all right. Actually, what I just doodled is right there. There it is. So this core here is the DNA itself. And then on this side, see how these are shorter? You see how these are all shorter? So what you're seeing here is this. You're seeing many, 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 many RNA polymerases doing their job. And as they read down this way, the RNA gets longer. Okay. And you can also see that as the gene is being transcribed, you can already see the spliceosome starting to do its job. Okay. So the second so here, if this is the DNA, this is the polymerase. The second that transcript is coming off, it gets capped. That protects it right away. And then the spliceosome grabs it. And then only after it falls off, you get the polyadenylation and the splicing. You don't have to know that level of granularity in terms of the stepwise stuff. Just understand, right? We have initiation, we have elongation, we have termination, right? That's transcription. Oops. That's transcription. How much room do I have to write here? Here. TX, that's transcription, initiation, elongation, termination. And then while elongation is occurring, we got capping, we have splicing, we have tail, cap, splice, tail, right? And then lastly, when everything's done, we export. All right. So the polymerase untwists the double helix about 10 to 20 bases at one time. So basically it pries open an entire twist. And you get about 40 nucleotides per second in eukaryotes, which isn't that fast. The average gene is thousands of base pairs long. So this isn't like neurons firing. Okay, this is more like from the time you wake up until the time you start feeling like a human. Well, for some of you, that's fast. The time you start drinking your coffee to the time you feel it take it into effect. Okay. I said that already. Okay, and then termination. There's just one slide on this. In eukaryotes, RNA polymerase reaches the end of the coding region and then it just falls off. The book goes into more detail than you need to know, and it is interesting to read, but I'm not going to hold you accountable for that information. In prokaryotes, there's so rho is a Greek letter. It looks like this. There's rho dependent and rho independent. You don't have to know any of the details there either. Just understand rho is a protein, and there's a terminator region. Alexa.
This is like a slide back, but you were saying that the RNA polymerase that like unwinds everything, is there like a topomerase involved there?
Oh, that's a great question. No, and here's why. When we open, well, so there's a bubble, right? You can think of it like a bubble, just like in our DNA polymerase. But the thing is we close it back up again, right? So we temporarily unwind it, and then it closes again. So we actually don't want a topoisomerase, right? If you remember in DNA polymerase, we unwind it and we want it to stay that way because we're making a new double-stranded piece of DNA, right? So the polymerase is more like a, think of a rug that has a marble underneath it, and the marble's rolling under the rug, right? So it's moving the rug, and then as it leaves, the rug settles again. Right, so the DNA untwists and then it goes back together again. So now there's a helicase activity, right? But you don't have to know any of that. Just RNA polymerase, think of RNA polymerase as the hollow enzyme of the DNA polymerase hollow enzyme with all the moving parts in it. You don't have to know any of it. Okay, so there's an enzyme that swoops down, pulls the DNA apart, and says, and is told, start reading over here, and it sees a U, or it sees a T, right? And so it's a here, I need an A. And then it starts building. You still look like you have a headache.
I guess like I'm just trying to like wrap my head around it conceptually. So I guess like the difference between like this process and like DNA replication would be like with DNA replication, you're like pulling apart the whole thing to duplicate it, while like this, like you're just pulling from DNA
here,
specific
here.
chunk.
Maybe this will help. Right, so this is DNA, right? Imagine sticking my thumb in here and then just moving my thumb along like this. Okay, now it's not entirely accurate because I can't redo it, but yeah, it's just well, maybe there is a topoisomerase, but now you're making me think a second about this. I never thought about topoisomerases and transcription. Oh.
Follow-up question to what you just did there with like the wire and everything. So like it'll run the whole length of the DNA strand. It won't just pull a chunk from it then?
So ask that again.
So like you're saying like the bubble, it's like moving down like the railroad track of like the double helix. So like is it going to go like the full length of the DNA, or is it just gonna like pull a chunk out?
Oh, it doesn't pull it. Well, when you say the full length of the DNA, do you mean the whole chromosome?
I guess just like the full length of like a double helix.
So the big picture view here would be so what I'm about to draw, this single line is double-stranded DNA. Okay, so let's say that the whole chromosome, right? Remember that chromosomes have those, the chromatin territories, right? Remember the whole histones and all that. So we're going to zoom all the way down into that. And so here's polymerase. And so what all polymerase is going to do is read along here. And so that bubble is inside of it the whole time. So as it leaves, right, like think of that as like the downstream and the upstream, right? So as we go downstream, we open it, and then right behind us, it's closed again. So we're not permanently making any changes to the double helix. We're just peeking in there and coming back out. Does that make sense? Okay, and that's for the topoisomerase. I do want to roles of eukaryotic topoisomerases in transcription. 2016. Oh, that's funny. this way. I learned this stuff a little bit longer than 2016. Topoisomerases, catalytic intermediate, blah, 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 blah. Here we review the roles of topoisomerases in mediating chromatin dynamics, transcription, replication. Okay, yeah, this is just a review article that was published in 2016. So it looks like topoisomerases do play a role in transcription. And that makes it, it's funny. The second I started moving my thumb along there like this, like I immediately realized like downstream of that, it's going to keep getting tighter. So you do need a topoisomerase there. Even though you're not leaving it that way, you're still pushing forward and creating a tension there. So yes, a topoisomerase would make sense there. I've never thought about it before because it doesn't play any regulatory role. So it's just part of the complex itself. Okay. All right, so elongation and then termination. So again, termination, the RNA polymerase keeps reading, it gets through a polyadenylation signal. All right, I want to make sure that's there again. Polyadenylation signal. It's like a word. There's a sequence in there, and that consensus site tells the poly A polymerase to bind there to make the poly A tail. Okay, so that's embedded there. That is part of the open reading frame. It's the last tail end of the open reading frame, no pun intended. Okay, the terminator, we only talk about that in prokaryotes. Eukaryotes don't have a specific terminator sequence. Okay. All right. Okay, so you just learned transcription. So we're going to draw a line there. Now we're going to do RNA processing, which we've already covered for the most part. Okay, so we have splicing, gets rid of the introns. We edit both sides, both ends. The 5' end gets a cap, the 3' end gets a tail. And then we export it. Okay, so you have to keep in mind, export is the last step here. So you'd say this: cap, tail, splice, export. You cap it, you tail it, you splice it, and then you kick it out. And the reason why I want you to keep those 4 steps in mind is when we learn regulation next time, each one of those steps can be regulated. For example, imagine preventing it from leaving the nucleus. Now you don't get a protein. Okay. Here's a schematic of eukaryotic gene expression. Okay, so in this, I'll very quickly show you. Here are the exons. Okay, so here's the DNA. Here's the primary transcript. You can see it's capped. It's got a tail. We're going to then splice these introns out. And they've done a little fancy job showing you their varying lengths and size. Doesn't matter. Okay, so these things, a lot of questions are, what do introns do? You can say they just get degraded and we reuse the pieces. Sometimes they play other roles in regulation, and I'll talk about that next time. And you get this mature transcript, which technically isn't really mature until it leaves, right? Just like your kids have to leave the house before you tell them they're grown up. So this is the 18-year-old, and this is the 26-year-old, the one that finally leaves the house. Okay. So this is just another overview of it. This is where, okay, this is the anatomy of a mature transcript. And there's a lot on this slide here. So let me give you a simple, I'll draw it slowly for you. Okay, so this is the mRNA. This is your 5' cat. This is your 3' tail. Okay. And here's your start codon. Notice I drew it not at the beginning. You see that? And then here's your stop codon. Again, notice I didn't draw it at the end. So this region right here, we can all agree that that's not going to be translated, right? We're not going to translate those parts. So those are untranslated regions. We're going to call those untranslated regions, UTRs. And this is the 5' untranslated region because it's on the 5' end of the transcript, and this is the 3' UTR. Okay, so untranslated region. These regions contain important sites. Okay, this is going to be where the ribosome binds when we do translation. This contains the poly A site for the poly A polymerase to bind. Now, the reason why we hand wave translation a bit is because we still don't fully understand all of this. There are translation factors just like there are transcription factors, but we're not going to learn about them. Okay, this is one of those, it's still kind of a black box. There's a lot more known about it than is in your book. There's a lot more known about it than is in my brain because I didn't focus on translation at all. I focused on transcription. It turns out this is just as highly regulated as transcription is, but we're not going to learn any of that. Right? We can all agree this is already information dense enough. So in here are all your code, right? That's your open reading frame. I mean, really, this thing, the open reading frame includes the start and the stop too, right? All your codons are in there for all your amino acids. Okay, so now let's go back to the slide, and this is in your book, so now you can understand this. There's your untranslated region for your 5'. There's your methylguanine cap. There's your poly A tail. There's your polyadenylation signal, which is AAU, AAA. You don't have to memorize that, but that's the polyadenylation site. And the poly A tail is anywhere from 50 to 250 amino acids. I'm sorry, nucleotides. So it's got a variable length. You can imagine the longer it is, the longer the RNA is going to hang around. Okay. Here's a schematic on splicing. This stands for small nucleolar, the nucleolus, right, inside the nucleus, small nucleolar ribonucleo. In other words, a molecule that's made out of both proteins and RNA, ribonucleo protein. Well, I should say the ribonucleo RNA plus protein, so a ribonucleoprotein. So small nucleolar ribonuclear protein. Okay, and RNP is the generic phrase, oops, not RNA. A ribonucleoprotein is a complex of RNA and protein. So the ribosome is an example of one, and the spliceosome is an example of one. Okay, so these are new things to you. You've learned about proteins, you've learned about nucleic acids, you've learned about lipids, you've learned about sugars, and now you learned about ribonuclear proteins. Okay, so it's like a glycoprotein, but instead of the sugar being the other part of the team, it's the nucleic acid, in particular the RNA. Okay, and so you can see here that the spliceosome is made out of small nucleolar ribonuclear proteins. You don't have to worry about lariats. Okay, any questions on that? This is from your book. And you can see more carefully now, those are those boundary elements I told you about earlier. The intron-exon boundaries are recognized by these small RNAs within the spliceosome. Okay. And this cutting occurs not from enzyme activity, but from ribosome activity. Which is pretty amazing. Okay, next slide here. This just shows you the pre-mRNA versus the mature RNA. So when you see pre-mRNA, your book calls it pre-RNA. I call it the primary transcript. The book calls it the pre-mRNA. It's the same thing. The numbers just tell you about how long everything is. Again, we're talking about +1 being the AUG start site. Okay, so you can see here Exon 1 is about 30 amino acids or 30 base pairs long. Okay, and then there's an intron, and there's another exon between 31 and 104. So do you see these numbers? These numbers are all basically bookmarks or page numbers. Okay, they're the base, they're the base number from the first. So you could see here, this transcript is 146 bases long, Alexa.
So where between like where that little dashed line is and the 5 cap, and then at the end where the little dashed line is in the poly A tail. So those are, that was the polyadenylation signal and the 5' UTR that are not translated.
Correct. This dotted line represents the start of translation, and this represents the end of it. And so everything past this is UTR. And everything past this is UTR.
And
Actually,
then,
you can see it right here. There you go. This right, this little red, this, or rather this box right here. There's your UTR. There's your 3' UTR.
so that
Okay.
does go down into the mRNA.
Yes. It was no. This whole thing is mRNA. Okay, so this is the pre-mRNA, and then this is the mature mRNA. Okay, the book says so. This just says mRNA, and this is pre-mRNA. So you could also make the argument that it's not technically mRNA until it's ready to actually get translated. I like the idea of calling it the primary transcript. It's hot off the press, and then you got to do stuff to it, and then it's matured. Okay.
And one more thing. So the stop
Yeah.
and start codon are still in that mature.
Absolutely. They're right here is AUG, and right there is UGA, or one of the other two. You got it. I just mentioned ribozymes. Ribozymes kind of broke people's brains a little bit, because up to this point, we assumed that proteins were the only things that catalyzed chemical reactions in our cells. It turns out that there are also, so already here's the thought, how you organize your brain's ideas, right? You have, in chemistry, right, we have something called a catalyst, right? They speed up chemical reactions without getting used up in the process. In biology, we have catalysts made out of proteins, which we call enzymes. And we have catalysts made out of RNA called ribozymes. Your book kind of does you a disservice by saying that ribozymes act like enzymes. It kind of suggests that enzymes kind of trump ribozymes somehow, when in reality, you would say ribozymes, like enzymes, are catalysts. They're both, so these are biological catalysts. A biological catalyst made out of protein is called an enzyme. A biological catalyst made out of RNA is called a ribozyme. DNA does not do this yet. I'm sure somebody will discover it does someday. But as far as we know, DNA does not catalyze chemical reactions. Only RNA can. Okay. Any other questions? All right, you guys still okay? Well, I wanted to get through transcription before taking a break. I think we're almost there. All right, here's a little thinking question, right? So why do we have both? Why do we have ribozymes and enzymes? Why would a cell have both? What's your answer? Think of evolution. Nobody's got a thought? Hold on a second. So the first cells on Earth were probably filled with RNA. RNA did everything. RNA stored information in a sequence-specific manner, so they were genes, and RNA catalyzed chemical reactions. So they did the work of the cell, and they stored the information of the cell. As evolution went on, DNA eventually emerged. And so the way I like to think of it, here's my quick story of it. RNA is an entrepreneur. You know, when you first start a business, you have to wear all the hats. You have to be the accountant and the marketer and the creator, and you have to go out and talk to customers, and you have to build the thing, right? You got to do everything. And nobody can do all those things at the level of excellence you need to run a successful company, right? That's how you start the company. Once you get going, you start hiring people. You hire specialists. You need an accountant to manage your money. You need a marketing person to go out and get sales. You need some, people in the factory to manufacture stuff, right? While you're in the background, you've got your hands in everything, right? Think of the CEO that bops around and kind of visits every department and knows enough to be dangerous and asks the right questions, right? That's a successful manager, somebody that understands enough of what your job is where they can talk to you intelligently about it. Okay, that's RNA. So RNA can store a little information, RNA can catalyze some chemical reactions, but it delegated the task of storage to DNA, and it delegated the work of the cell to the proteins. But it always stayed there in the background, and it's got its hands in everything. That's why RNA is kind of a molecule you can't quite wrap your head around, because it does it all, and it's a mess. Okay, all right. All right. Here's the last concept. I've introduced the idea before to you of protein domains. All right, consider a protein. Let's just consider a hypothetical protein that here's DNA. Let's consider a positive transcription factor like E2F. So E2F has a region of the protein that is a DNA binding domain. Okay. And then, so let's say that DNA binding domain has some alpha helices and some beta sheets and whatever. Let's do this. Okay, so there's pair alpha, it's called a helix loop helix. This is beautiful little motif that sticks to the groove of DNA, like a pair of scissors. Where's my DNA? Right, so you got the groove of DNA. Imagine a pair of scissors that can slide right into the groove like that. Okay, so that's what that is. We'll call that a DNA binding domain. And then as it goes on over here, let's say there's a little beta sheet and maybe another helix like this. And so we're going to call this the activation domain. And the activation domain is what, say, binds to some protein like RNA polymerase. This making sense? Okay, so proteins, you can see, so this protein right here, right? If I was to just grab it now, let's see if I can. All right, so this protein right here, oh, perfect. All right, so in reality, right, it's one protein, one continuous polypeptide chain, but there's two regions of it that do different things functionally. So that's what a domain is. A domain is this functionally relevant part of a protein. So I want you to be familiar with this concept of a protein domain. Okay. Now, here's where things get really awesome. When you look at a gene, so this hypothetical gene that I just made, here's exon 1, and here's exon 2. You see where I'm going with this now? Okay. This exon all by itself will give us that DNA binding domain. This exon all by itself will give us that transactivation domain. So in other words, these are like Lego building blocks. Imagine taking an exon from one gene that binds to DNA and mixing it with a part of another gene that does something different. Okay, so these right here are the Lego building blocks of evolution. That's the way to think of this. Okay. So imagine for the same protein, imagine that I had the spliceosome working on it. Right, so here, I'm just going by what I doodled up there. So there's right, this is intron 1, I should say. Let's make the intron yellow. All right, so there's intron 1. Okay, now when I splice that out normally, right, you would get this, right? But what if I spliced it where I ignored, let's see, if I spliced it here, but then I ignored this splice site. I just ignored it. Then the loop would look like this. And let's see here, just for argument's sake, let's say there's another exon over there. So I can do this. Okay, so here's exon 1, here's exon 2, and here's exon 3. And so here's another intron. So yellow's all intron. Okay. So this here. So there's that. There we go. Okay. So in other words, when I spliced this, I ignored exon 2. So my mature transcript now is only going to have exon 1 and 3. Okay, so this is called alternative splicing. And this is how you can go from one gene encoding information to make several polypeptides. So the one gene, one polypeptide model isn't true either. Okay, so here, I'll give you a simple, another schematic of this. So here's the gene, and here are the exons. Okay, let's say this gene has 7 exons. So you can imagine a transcript that has just exon, well, actually, let's make it even simpler, just exon 1, or exon 1 and 2, or 1, 2, and 3, et cetera, or mix and match them. Imagine I had 1, 2, 4, 6, and 7, or 1, 2, 3, 4 by itself, or 1 and 7 by itself. Okay, just pick, pick where you're going to splice. And you can get a whole family of proteins now from one gene. And so this family of proteins is called a family of isoforms. And we usually name them with Roman numerals. So for example, there's a protein called PML, and there are 7 isoforms named PML1 to PML7. And they all have slightly different functions. Okay. Raise your hand if that made sense. Okay, sort of. So Alexis, ask me a question.
So for each of those proteins, they're each a different domain.
Yeah, there's different. Yeah, that's a good way of thinking of it. They have different domains. So for example, maybe PML1 includes the DNA binding domain, but PML3 doesn't. And so PML3, because it doesn't have its DNA binding domain, but it has everything else, right? It's going to still bind to RNA polymerase, and now it's sequestering it. So it acts as a negative regulator. Okay. And you're starting to see the cracks into how insanely complicated things get. And we're still not anywhere near the actual level of complexity. Okay, which is why I have to keep telling you guys where the level I want you to know, right? So understand alternative splicing, understand splicing, understand protein domains, okay? And you're starting to now become proficient in molecular biology. That's what all this is. This is molecular biology. Okay. All right, cool. Cool. And so this is where we're going to stop for our break. The idea is called exon shuffling. I just learned this later in life myself, and this blew my mind. You guys remember crossing over? Remember crossing over in Mendel, right? You can get your recombinants and all that. So I want you to now take everything you already learned, but instead of talking about or considering different genes, I want you to think of a gene with 3 different exons, 3 different domains, A, B, and C. Okay, so on this slide here, right, you can see, so the blue one represents, whatever you got from mom, and orange would be whatever you got from dad, right? So there's your genotype, capital A, capital B, capital C, lowercase a, b, c. But these, instead of these numbers or letters corresponding to genes, they're the different exons, right? So capital A gives us a circle, capital B gives us a square, capital C gives us a star. And so the protein's going to look like this. Okay, the lowercase a version gives us a heart, the lowercase b gives us a triangle, and lowercase c gives us this 6-sided star, sun. And so these proteins look completely different from each other, right? So imagine crossing over occurred here. Imagine crossing over did this. So now we get capital A, capital B, lowercase c, and we get lowercase a, lowercase b, capital C. We get completely different proteins now. Okay, this would be like taking your kids' toys and popping the heads off of one and putting it on another, and taking the gun from that army guy and giving it to this guy, or taking, putting this shirt on this doll, or whatever. We're mixing and matching parts to make new things. And it's called exon shuffling. And that's the real reason why introns are so long too, is to make sure that crossing over can occur between them. That's another reason, I should say. Okay. All right, let's take a 10-minute break, cleanse our brains, because I know this is a lot of information and you're getting hit with it fast, but I don't really have any control over this.
Do you know how many more chapters there are that we need to learn?
So this is 17.
17.
So there's 18. Okay, and then just parts of 20. 20 is biotech. We're skipping 19, that's viruses. Okay, and then this chapter here, 17 and 18, is just genes, right? So gene expression and gene regulation. And so all of them it's all interconnected, right? If I was to write a book, I would probably put those in the same chapter and have 2 chunks, expression then regulation. So it's not as much as you think to get to the test. I know. Okay.
Thank you.
Yeah, and focus. Remember, you got the study guides. Let those be your beacon to guide your way in the study. All right, let's take a 10-minute break, and I'll see you guys in 10. All right, I'm back. Okay.
For this exam, like the next one coming up, what chapters is it?
Sorry, hold on. You just broke up, Tori. Say it again.
For the chapters for this exam coming up, it's chapter
Chapters what?
15 to 18, right?
Yeah, no, 16. 16 is DNA.
Okay.
16, 17, 18, 20.
Oh, okay.
Okay. So the rest of this stuff we can skip here. Okay, here's RNA splicing. You can see the different domains, right? Same idea there. So the number of genes we have, right? So when we talk about all our genes, we talk about our genome, right? So there's our genome, and then genes make proteins, right? And so we have what's called the proteome. So the human proteome is way bigger than the human genome. We don't even know how big it is still. There's estimates. That's all we can do. Okay. I've already explained all this. New combinations of domains give us new proteins and possibly new functionalities. Right. All right, translation. Oh, I have a question for you guys. When I said earlier, do you want to do PowerPoint or do you want to do whiteboard? And most of you seem to do the PowerPoint. Is there something I can do better in the whiteboard to make that better? Because in the past, I've had students want to use the whiteboard more. So seeing that shift, I'm just curious to know what your thoughts are. Why one's better than the other?
Well, I like PowerPoint because I feel like it's a little more organized and it, like when you do whiteboard, you kind of jumble around, but
Yeah,
like,
that's that. I was
there's
thinking that too.
this.
Okay, so that's usually a sign of me being tired when I do that. So that's probably what's going on there. So let's do this combination. We're gonna do translation now. So I'm gonna bot back and forth, and I'll try to stay organized here. All right, so translation. Key players, right? We got the ribosome. The ribosome consists of the small subunit, the large subunit. So you can look at the small subunit, it kind of looks like a boat, and the large subunit looks kind of like a half hemisphere. And really, you can think of it collectively as, if you've ever watched South Park, you know, Terrence and Phillip, the way they draw Canadians, that disembodied head. And you can see that the large subunit of the ribosome has 3 grooves in it. And those grooves are the E site, the P site, and the A site. You could think of the Environmental Protection Agency, EPA, the EPA. E stands for exit. P stands for polypeptidal. And A stands for, so it depends on who's talking and what book you're reading, but you can think of A as the aminoacyl site, or you could think of it as the acceptor site. Okay, so that's the ribosome. key player 1. Key player 2 is your mRNA. You can call it the mature transcript if you want. Key player 3, we have our, let's call them our amino ACL tRNAs. And then lastly, we're going to have something called the release factor. Okay. So those are our key players: the ribosome, the RNA, the tRNAs, and this thing called the release factor that's going to end the game when we're done with it. Give me 1 second, guys. I have to respond to something here. So you keep here. Keep writing that down. All right. I'm gonna have to go back to that again. So anyway, there we go. Okay. Any questions yet? Feel free to interrupt me as I'm explaining this with your questions. Okay. All right. So translation steps. Step number 1, initiation. Initiation begins when we have, I want to draw straight lines here. And here, we're going to have our AUG site. And let's then say that the second codon here, let's call that CCC. The third one here will be GGG. And the fourth one here will be ACG. Okay. We have our 5' UTR. We have our 5' cap. Right. Okay, now I'm going to take this whole thing and copy this, because I'm going to need this again a couple times. Okay. All right, initiation. The first thing that happens is the small subunit binds to the 5' UTR. Put it over here. All right, so you can abbreviate, so it looks like this. I use purple. Okay, so there's our small ribosomal subunit. And so you can picture this as able to slide along here. Okay, so we're gonna bind it here. It's gonna slide along until the AUG is kind of smack in the middle like that. The next thing that happens is the initiator tRNA. Initiator tRNA binds the start site or the start codon. Okay, so we can, let's use kind of a fuchsia here. All right, so here we go. And of course, this is going to have the anticodon, you know, whatever, UAC. And it's going to have on it the methionine, that amino acid. Okay. And then the very last thing that happens, number 3, is the large subunit binds. And here, let me draw these in a way that I'm just redrawing it. You don't have to, you're not doing anything wrong, but I want to make this internally consistent here. Okay, there we go. Okay, and then so here's the large subunit. And again, remember there's an E site, a P site, and an A site. So we can even color code those as kind of like red, yellow, green. Okay, so do you see how the initiator tRNA is in the P site there? At all times, the P site is going to be the keeper of the polypeptide. That's why they call it the P site. So that's it. That's initiation. Elongation. I'm not going to write this. I'm just going to show it to you. We're going to take this whole ribosome. Let me see if I can do this better. Let me pull out. So I can pull this apart. Okay. Oh, I grabbed the U. I didn't mean to grab the G there. Put that back. Okay, so there's the ribosome. I'll tell you what, let's get rid of the colors to make it easier to see. Okay. All right, so we're going to take this ribosome. Okay, so you see I can slide it along. So I'm going to move it from, hold on. Oh my God. I'm going to take that out. So watch this. I want to take the ribosome itself. Okay, so I'm right there. See the AUG's in the middle, right? You see that? I'm going to move the ribosome 1 codon downstream. Okay, but see the tRNA here? The tRNA was sticking by hydrogen bonds to that start codon, right? This is a hydrogen bonding. So the tRNA didn't move. The ribosome did. Okay, imagine standing in front of a train that's coming onto the platform. You're standing still and the train's moving and the doors open in front of you and there's too many people there. And so what do? You wait for the train to slow down a little bit or to move down a little bit more, and then the next set of doors opens. Okay, so the tRNA is not moving, the ribosome's moving. Okay. And so so right now that RNA is sitting in the E site, right? That's the E site there. Oh, it didn't move yet. I'm sorry, guys. And you guys are very focused and copying all this down, and I just realized before we do the move, let me just undo this here. Okay, so before we do the movement here, let's put this back here. Okay, so the A site's there. And okay, so we haven't moved yet. So right now, that initial tRNA is sitting in the P site still. Okay, nothing's moved. Before the ribosome moves, another tRNA comes in. And this tRNA comes along with its amino acid. And this is in the A site. Okay, so there's the EPA again, right? So do you see how the two tRNAs are sitting right next to each other? One's in the P site and one's in the A site. Okay. Now the ribosome is going to move one codon down. And while it does that, it is going to transfer this amino acid onto that amino acid. It's going to catalyze the peptide bond so that we can literally just take this little blue or green dot and put it here like that. You see how I transferred it over there? So at this moment in time, do you notice how the tRNA that's sitting in the exit site is now empty? Right? It's like a bullet with it's the empty shell. And because it's in the exit site, what happens is this empty tRNA leaves. It exits stage left. In the meantime, notice that this tRNA is now the keeper of the growing polypeptide. There's the P site. Okay, this would be like you wearing a hat. Let's say you're wearing a hat. Somebody else comes along with the hat and puts the hat on top of you. Okay, and so now you're wearing two hats, and somebody else comes along with a hat, and you take your two hats, you take them both off and put them on the other person, and then you leave. And then another person with a hat comes along, and the person with the three hats takes all three of their hats off and puts it on the other person, right? So the idea is you're not just adding one at a time, you're literally moving the whole chain. Okay. And so now do you notice that the A site is free? So the next tRNA comes in here. And we just keep doing this. Okay, any questions at this point?
Where did the tRNA come from?
They're floating around. Okay, and the only ones that stick for any length of time are the ones that have the proper base pairing. They have the right anticodon, so they can linger because they can stick like Velcro.
So they just like kind of exist within this region, but like with the correct base pairings or not?
Yeah, they'll bump, like, let's say that like all the tRNAs are there, right? So the ones that don't have the right anticodon, they come in and bump around, but they don't stick. And it happens at thousands of times per second because we're at the molecular level. Everything's moving super fast. So the only thing that actually sticks there is the one that has the right specificity.
And then when they exit this complex, did they lose those base pairs?
Say again, Alexa?
When they exit this complex, are they going to lose those base pairs that they came in with?
No. So those base pairs are integral to their composition. Let's go to the slide for a second. Let's look at let's see if there's a cartoony version of a tRNA here. Yeah, okay, here's tRNA. Here's the player. All right, so the sequence of tRNA looks like this in 2 dimensions, right? It forms this two-dimensional structure. There are this base pairing that occurs. Looks kind of like a 3-leaf clover, right? And then what we do is we take that 3D and then we kind of fold it up into, really, it looks like a gun, like a ray gun. It's got like you can picture your hand here holding it and it shoots that way. And then over here where the bullet comes out would be where the amino acid is. And down here's the anticodon. So you see how the anticodon is integral to it. It doesn't lose its anticodon. It stops sticking. I think that's maybe what you were talking about here. So this tRNA here leaves right? This falls off the base pairing. The ribosome kicks it out. Okay. All right. Other questions?
I have a quick one. So every time another amino acid is there, it moves it down by a code?
Every time the amino acid,
Like an amino acid is added, it moves down by a code?
yeah. So basically, once the tRNA hands its amino acid to the next one, the ribosome shifts off so that they can leave and the new one can come in. Okay. And so we keep doing this again and again and again, and eventually the ribosome keeps moving this way. The tRNAs keep coming in with their amino acids, and over time we get this polypeptide that emerges, and then it starts to fold into its alpha helixes and beta sheets and all that until we come to UGA, the stop codon. Okay. When the ribosome hits the stop codon and the ribosome is sitting with the A site in the stop, let me say that differently. When the stop codon is in the A site, right? So when we have this thing is here, when the ribosome has the A site occupied by UGA, by the stop codon, there is no tRNA. Instead, there is a protein. I don't like red. Let's use a lighter color. You can see better. Let's use yellow. There's a protein that binds here called the release factor. The release factor binds and it causes dissociation of the entire complex. It basically breaks the ribosome apart and it pops the polypeptide off. Does that make sense? I hope. Okay, let's look at a movie now. So that first link, I have to delete that. The first link doesn't work anymore because it's a Flash animation. Flash doesn't work anymore. Neither does that one anymore. These old websites didn't update. Hold on, I know this exists somewhere. Virtual cell animation translation the movie. Let's see if there's a migrated. No, not that. Ugh. The ones from the textbook drive me insane. They're here we go. BSAC. Flash animation. Does anything use Flash anymore? Why? I thought that you could still use Flash in something. That's really frustrating.
When they shut down Flash Player like
Advanced
past
version of
December?
it. What'd you say?
When they shut down Flash Player like this past December.
Yeah, I forgot they did this virtual cell animation collection. Oh, that's nice. Yeah, all right, whatever. Oh, there it is. Okay, hold on. It must be on YouTube somewhere. Full cell translation. Yeah, there it is. Okay, this is the original video that I wanted to show you. Let me share my screen here. All right, can you guys see the screen? Oh, let me share the video sound too. Okay. The music is really nice and cheesy. All right.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site.
All right, so they called it the peptidyl site. It doesn't really matter. The idea is the P site keeps the polypeptide. You guys following this still? Is this making sense? Okay.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon.
All right, pause it to absorb this here. So here's your start codon. Here's the anticodon. Here's your initiator tRNA. There's a methionine. All right, so right here is the, can you guys see my mouse? Make sure I can. Okay, so that's the P site. All right, the A site here, UUUAAA. Here's the second amino acid. And so what's gonna, it's basically these two, the tRNAs are shaped like this, like they're spooning, like they're laying next to each other. Okay, and so that the 2 amino acids are positioned precisely so that we can catalyze this bond right here. So watch this.
The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
Okay, so you see how the entire polypeptide is transferred, like a bunch of hot dogs linked on a string. So that's the elongation.
These are the basic steps of elongation.
And do you see how the ribosome itself is moving while the tRNAs stay still? They're staying still because of the hydrogen bonding.
As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
And that's it. Okay, so 100% of proteins are made this way. Now you can go back to your cell biology chapter and remember ribosomes insert themselves into the rough ER. Ribosomes are found in the cytoplasm floating around, or ribosomes are in the mitochondria. And you also remember that there's ribosomes that prokaryotes have that are smaller than the ribosomes that eukaryotes have. And the ones in our mitochondria are the smaller ones, right? So our mitochondrial ribosomes look just like bacteria ribosomes. Okay. All right. So it's 1 o'clock. Let me just I just want to wrap this up now since we're at 1 o'clock here. All right, so let's see. That's it. Anything? So I taught you all this already. You can just review these slides now. Yeah, we talked about this. Is all really. I threw all these slides in here just for your own study after we reviewed all that. Okay. Okay, so the very last thing I'll say at the end of this is that once a protein is manufactured, once a polypeptide is manufactured, that's the beginning of its story. All right, so there's a whole world called post-translational, right after translation, post-translational modification. You guys have already learned one, phosphorylation. Remember, some proteins are phosphorylated, right? Remember G protein coupled receptors. Remember that, or adenylyl cyclase or G proteins. And remember the idea of phosphorylating a protein to activate it. So that phosphorylation event is considered a post-translational modification. Okay. Other proteins will need to be cut up to be activated. So for example, some enzymes are synthesized, but they're dormant. And so you cut a piece of them off to activate them. Okay. All right. And so at the end of the day, we can now say what a gene is and what a mutation is, right? We used to say that a mutation was something that changed a phenotype. We can now say it's a change in the sequence of a gene. What's a gene? It's a sequence of DNA that makes a piece of RNA. So that's that. And one thing we're not going to cover in any great detail this semester, because we literally just can't fit any more into the class, is this notion of RNA inhibition. I will mention this when we do regulation next time, but we're not going to spend any amount of great time on it. Okay, and that's that.
Is this the
So
end of chapter 17?
this is the end of 17, yes. Okay, so next time we'll do chapter 18. And I'll teach you the lac operon. Okay, I would encourage you guys to get as far as you can on your own. We have a big gap here, right? So feel free to ask me questions between now and Monday on your studying on the lac operon. Even though I didn't lecture on it yet, I'm totally okay teaching you guys over the weekend if you want. If you're sitting there reading and studying on your own and you pick my brain, I'm here. Okay. Put this link in the chat. Yeah, I'll definitely, I can do that right now. So here, I'll put this in Zoom meeting. Let me stop sharing my screen so I can use my computer here. All right, let's see, Zoom, and there we go. All right, so that's the translation video. There are a ton of awesome animations out there for all this stuff right now. You guys have access to stuff I could only have dreamed of when I was in your seats. So you'll have a much quicker, better way of understanding this stuff than I ever did, which is kind of nice. I'm a little jealous. All this was when I was in your seat, all this was just static pictures and imagination. There was no animations at all. There were no computer, anything. It was very hard to understand.
Can I ask a question?
No. Yeah, of course. Go ahead.
Just about, like, I guess the course. Do you think we're like gonna stop failing everything? Do like your like our exams and stuff? How
So wait, hold on.
averages are so
Remember,
terrible all the time. And I'm just curious, like, is that something that always happens? Or do you think it's
Yes.
because, yeah, it
so
always happens? It's not
well,
just,
no. So keep them. So you're approaching this from the perspective that raw score you get is your actual grade, and that I have to then modify it,
I
right?
know.
And so that's the wrong way to look at this. The right way to look at this is these tests aren't perfect because we're sitting here in Zoom and we're sitting here in a pandemic, and I've never done it this way before and you've never done it that way before, and we're all trying to figure out how to make this all work, right? So it turns out the people that have been teaching for like 20 years, right? People like Professor Korn, she has honed her craft down to the point where she knows exactly the kinds of questions to ask and how much time it takes and how it covers lectures, to the point where her exams would give her a number that she still has to adjust a little bit. Okay, Steve Gosshorn, we talk all the time. He's constantly, every teacher has a technique to process the raw outputs that students give. Okay, so for these classes, the other struggle is you guys are this broad spectrum of student, right? You guys aren't all 18 with a specific SAT score and applied to the college and got accepted, right? If you think about a classic freshman class at Temple or Drexel or wherever, all those students are kind of homogenized, right? They're all about the same age. They have about the same high school GPA and experience. And so you're going to see a nice bell curve in that kind of situation with any, so the average will be a C. And I mean, that's, you get what you said. What you see is what you get. In community colleges, because it's open access and anybody can register without the need of presenting things like evidence that they have prior learning and all that. You know, you guys have to take placement tests, but that's about it. You get what's called a bimodal distribution. So you can't see my screen anymore. A bimodal distribution in statistics, the bimodal distribution. So here's your bell curve. Okay, bimodal looks like this. Basically, when you do, and I see this a lot in most tests. Let me go back to this. I can't see you guys. You have people that do really good and really bad, and there's very few in between. And so when you have bimodal distributions, your averages sometimes get a little skewed because those bad scores are sometimes in the 30s or 20s, right? So when you see that average, whatever it is, you have to keep in mind that that average is all equal, right? So and people who get zeros on it because they didn't take it, sometimes that gets averaged in depending on how I input it. So what I have to do is I have to go in with a fine-tooth comb with these tests, and I have to find the questions that, for whatever reason, weren't faithful to assessing you. In other words, that question is not a fair way of me answering the question on my head, did the student learn anything? Right? That's what tests are. They're assessments. An assessment is, did the students learn the outcome of the class or the outcome of whatever the lecture was? So no way of grading is perfect. All right. So one other way to do this is give you a bunch of multiple choice and then a bunch of short answers or essays. Right. And when you do it that way, your essay questions tend to be higher scoring than the multiple choice. And so those rescue the lower multiple choice. Okay. When I was a freshman, we were terrified. We would have like 100 question multiple choice, and it wasn't curved, it wasn't adjusted, it was nothing. And after your test, you would go up like 2 or 3 days later, you go over to the board and you'd see your social security number. This is back in the 90s, and it would have a number next to it. And that's what you got. It was 87, 92, 47, whatever. And that was that. And so our freshman biology class, we started, all the students kept saying that freshman biology is the weeding class. It's so hard. Everybody's doing so badly. When the reality is there's so much to it. It's so detailed. And so we have to make these kinds of adjustments. So Taylor, you're doing fine. You're not doing, stop looking at the raw score, as what I want to say. So the adjusted score is what me, the professor, says the student earned. Okay, those aren't free points. Those aren't me giving you anything. Right, what I'm giving the class because of the pandemic is I'm bumping all your grades up that half letter grade. And that's because of the chaos that this is. But those adjusted scores, when you go and take Bio 2, for example, and I've done this before, I look at Joanne Korn's, I periodically ask her this, and I probably have to ask her this again. I haven't done it in a while. I'll say, like, so how are my students doing in your class? You know, the ones that I like, so you're going to go and take Bio 2 at some point, or some of you are, and some of you take her. And so she always says they do fine. And she grades completely differently. It's a completely different class, right? My litmus test is, am I preparing you guys for the next step, which is Bio 2? And the answer to that is yes. So does that answer your question?
Yeah, thanks.
Okay, yeah, so chin up, you're doing good. You are.
No, it just doesn't feel good.
I know. And that's, so really what I would probably rather do, and this is what some people do, is they hide your final grades. You don't know how you did on the test until I give you that adjusted score. And I actually used to do that. So if we were in person, right, the way tests would work, you take the test on Monday, right? And then on Wednesday you come in and I'd hand you a piece of paper, and that piece of paper would have a number at the top. 87. And you'd be like immediately, oh, I got an 87. That's pretty good. And then you look at your raw and be like, oh, if he didn't add points, I would have gotten a 72 or whatever. But you would have seen that second. But because we're in Zoom and we're in Canvas land here, you guys see that, you see the raw score immediately. I could hide that. And maybe I should do that next semester for psychological reasons, not logistical ones. Okay, so imagine you saw your final grade before your raw grade. Would you feel any different?
Somewhat. I mean, they're still just not good. You know what I mean? Like, they're okay. It's just, I don't know. I just wish, I don't know. Yes, I would.
Let me
better.
tell you another thing.
Still
Okay.
not where I want to be. Like, I just feel like no matter what I do, like I try so hard and it just reflects so little and it just makes me upset. So I was just curious if like,
Is it, and this is the test specifically.
honestly, it is everything. If I'm
It's
being,
the quizzes?
oh yeah, I do terrible on them.
Or are you talking about the homework and the labs too?
Oh yeah. Well, the Labster is different because at least I can do it again if I do bad. But like seriously though, like
You can do the homeworks again. You can redo the homeworks.
it takes me 3 hours to do that.
Yeah, I
Once it's
wouldn't.
done, it
I'm
needs
not
to go
suggesting you
away.
should do the. I'm not suggesting.
I just have a lot of like there's just so much, you know, like I try and write down the notes and study the notes. I read the textbook, I try and study that. I do the homeworks, I try and study that, and then I still do terrible on everything, and it's discouraging. It's just discouraging. But
I feel you, and I'm sorry that it's been that way. Jordan, did you put your hand up there, or were you waving goodbye?
Oh no, I was clapping because I was. Like,
oh, you were agreeing. See, so Taylor, you're not alone. You're not alone.
And I think that's the biggest thing. You do feel so alone because we can't even like, you know, interact with one another. Like I have like a lab on campus for chemistry and it was just so nice to like work with another human being and something and be like, okay, wow, like I'm not crazy for feeling how I feel because you feel stupid and I just hate it. So I didn't know if that was like something that just happens or probably just another terrible thing.
So well, that's
online.
That's a common feeling in all first year science courses. Science courses, especially the first ones, you know, whether it's physics 1, calc 1, chem 1, you're not used to thinking like this, right? This is different than English and psychology and sociology and all that stuff. It's think of it like martial arts in some ways. Think of it like you're taking your brain and you're hurting it and punching it and training it to think like a scientist. And that's not something you do in a day or a month or a year. It's slow and it's painful. Think about it. If it was easy, everybody would do it, right? So what you're feeling right now is the same way somebody would feel when they first started training for something that's hard and they're new, right? So, okay, there's a new show on Netflix I just finished binging this weekend called Shadow and Bone. Have you watched that yet?
I started it.
It's so good. What do you think so far?
It's really good. It makes me want to read the books because like I know there's more detail than the show. So.
And so it turns out the two plots for the two characters are actually two separate books and they merge them together for the TV show. But before I go too far on that, there's the main character in the show, Taylor, is this girl who discovers she has this latent talent. And she has to be trained to use this magic. And she's around all these people who have been training since they were like 3 years old. Right. And so they're all making it look super easy. So she's like, I can't do anything. She's broke. She's like, I'm useless at this. And then eventually she gets the hang of it. Right. So you're feeling growth pangs or these are growing pains as a science person. And it's totally normal. I felt that way. I still remember the feeling I felt in organic chemistry. I took that first test and it kicked my ass. And I thought I knew it. Okay. So
yeah, I guess it just doesn't really feel like growing. Like I see it when I'm like
you're learning. See, I
know
your
I
questions
am, but
tell
it
me
doesn't
you're learning
reflect. It doesn't ever like reflect in my grades. You know, like in my knowledge, yes, but then I don't know. What'd she say?
Oh yeah, I'm reading. So I just want to be able to pass with a 70 without a curse, and it's like impossible. I completely agree with you, Taylor.
Sorry.
Yeah. So first of all, you're gonna you're gonna get your 70, I'm sure, Tori, without, because again, remember,
It's so hard because like I study constantly and I feel like I get absolutely nowhere. Like
Yeah.
it's
that's not true. You guys are
But
all,
it feels like that.
yeah, and then
okay.
like the last exam that we just took, I tried studying like a different way to
Yeah, I
improve
felt
my
so
grade.
good about it. I went to
Worst
bed
one.
so ready.
That's the worst score I've ever gotten. So I was like, okay, that's like not a way to study. But like,
So there's a whole nother side to this, and that is my side, right? I have not perfected assessment yet, and especially over Zoom and teaching remotely and how to do all this. I don't think I'm doing it the best way either. So the thing is you guys don't know any of that, right? You guys come into a class, there's a guy teaching it, and he's doing his thing, and he's holding you accountable to all his work. And you know, I'm figuring things out too, right? This is my 3rd year teaching this class, right? There's people that have been teaching this class for 20 years. So I'm still a noob myself.
Yeah.
And I feel how you feel, Taylor, after I see those averages as the raw scores. I either get excited when they're high, and I feel like I failed you when I see those very low scores. Like, what am I doing wrong? That's my first question, is where did I do wrong? Not why are my students not working hard? I know you're working hard, right? That's not the question I have for myself. Is, am I asking them, are the questions too hard? Am I not giving them enough time? Am I not teaching them well? Should I do more of this active learning stuff? That's what I'm transitioning to. I'm trying my best to get to the point where instead of lecturing, I do a little lecture and then a lot of puzzle solving. The problem is a lot of this stuff is foundational, like translation, like just go through what we just did today, transcription and translation. How do I do a mini lecture on that and then ask you a bunch of questions? I don't know. Maybe somebody else has an answer to that. I'd always be open.
The thing is, all of this takes time to understand. So that's why, at least for me, like when you ask certain questions and stuff, I'm still like chewing on the definition of these things. And then, you know what I mean? Like, it's just a lot. And it's nothing, like, I personally don't think it's anything you're doing wrong. It's just the material, and it's how much there is and how in depth it is. And that in itself is discouraging, because it's like, how am I ever going to understand, you know? And then it's just like this whole like ripple effect of just.
And the other problem is your earlier education failed us, right? Like, in an ideal setting, I'm teaching you college level biology after you've learned high school level biology, and like for real, like transcription and translation and gene expression and just the basics of metabolism, what enzymes do and how things stick together, and collision theory. And that should already be there, but it's not, in my experience. Most students,
I took 2 years of biology. I took bio 1, I took bio 2. I thought maybe I would be prepared for something, like always like honors level 2, you know, like graduated with like 7 sciences. And it's like, I thought I was ready, and it doesn't, like,
and now it's like, I'm getting my ass kicked.
I'm taking chem as well. And it's just the same thing everywhere. I was top of my math class, and now I like can hardly do math. Like, it's terrible. So it's not just this, it's everything. It's like every class. I don't know what it is. I want to.
So one idea,
well,
you might have had an idea earlier. Remember how you said like being around people?
Yeah.
Okay. So this is a personality thing. When I was in lab and I would get an experiment back, I would get data back, and I couldn't understand it. I'm like, this makes no sense. I could sit there by myself looking at it and you know, taking notes and just thinking about it and just getting nowhere. And the second, you know, let's say I was alone in the lab, and then one of my friends would come in and sit next to me, and I'd say, can I show you this for a second? This is driving me nuts. And I'd be like, blah, blah, blah. And the second I started explaining it, I'm like, oh, like I just needed another brain to bounce my ideas off of.
No, you should see my lab notebook. Like I was working with this one kid that was next to me. Thank God we didn't get yelled at for like sitting too close to actually see each other's notebooks. But he was like, oh my God, you wrote so much. I'm like, I have to. This is the only way I can understand anything. And he has so little. And so like, it was just so nice to talk to another person about something.
So maybe you guys should, I mean.
That was the other thing, like, my husband is taking your class next semester, so
Don't tell.
So he watches me do like all my work, and he's like, oh my God, you're doing so much. And I was like, well, I'm online, like you're gonna be in person probably, and it's probably gonna be like a lot easier.
Tell him not to get his hopes up.
Well, he'll have like more people to talk to about it at
Yes,
least.
yes, that part is absolutely true. And we'll see. So what's his name, so I know when I get him?
Don't talk.
William Dresnut.
William. Okay. Is he William
He's
or
in
a
your,
Bill?
like, Will.
Will. Okay.
He's in your like 2 to 5 class, I think.
Okay. All right. I'll keep that in mind. So yeah, you guys could be meeting on Zoom together. I don't know if that would help, Taylor. I mean, I know that like Alexa and April. Well, you were there. You did it too. So
Yeah, it was Sammy,
you could-
Julia, me,
yeah.
Alexa, April. We all did like a miniature study group.
So
And,
are
and
you
wait,
guys?
Taylor, you did come in for like a second and then you left.
Yeah, I was at my boyfriend's house and he had to go take a call. So I was like, you know what? I'm going to join and see what we're doing. And everyone was just like crying. And I was like, I got to go. I can't do this.
Yeah,
Like,
we
everyone's
were
struggling.
That was a fun night. We were on that call until like 1 a.m. because Dr. Ippolito came in and then we just like, it was a whole thing.
I just like with Zoom, I just like, I don't like, I feel like definitely like the breakout rooms and stuff like that one-on-one with just another human being like of your choosing, you know, like when you're in a classroom and you're like, hey, I want to work with you, and then like you guys work together on stuff, and then like you lean over to another group and it's like, hey, like how are you guys doing? Like in Zoom, like literally only one person at a time can talk or you don't understand what's going on. So it's hard to work with other people over Zoom. It's hard to understand what other people are thinking, and if you do, it takes forever for me to say what I'm thinking and then you to say what you're thinking. It's just I don't think it's you. I just feel not good about all, and I just needed to stop. I just needed to stop.
I needed to
I just
stop.
need the semester to end, and it won't end, and I just want to cry a lot.
I'm burned out myself. I understand. I am definitely suffering from Zoom fatigue. Because you know I do this whole thing again right after this class,
Yeah.
and then tomorrow morning I do it again, and then Wednesday today's Wednesday, so Monday and Wednesday it's 8 hours of this basically. Tuesday and Thursday it's half that, so it's about 20 to 25 hours a week of Zoom for me. I'm so done.
It hurts, right? It
So-
hurts.
It hurts. It really does. So,
Yeah.
but so I don't, so I've been with you guys now for a couple months. Like I know you guys pretty well. I hear your questions. I see your work. I would say all you guys are doing great. You really, I know you don't think it, but so I'm telling you as your, you know, whatever, you're Mr. Miyagi, your trainer, you guys are doing good. You guys are meeting my expectations, right? And particularly the questions you guys asked during class. That's to me the most telling of how the learning's happening, because your questions tell me where your brain is in the relation to the material, right? Like, so earlier when Alexa asked me like, when you're like, is this a stupid question, DNA or RNA? Like, no, I see where your brain is when you ask that question. And so I'm able to then pivot and correct it, right? Like imagine it was martial arts and you're throwing a punch with your thumbs up here instead of over here. All I have to do is walk over and be like, put your thumb here, and boom, there you go. Go ahead, Alexa.
I was just gonna say, like, sometimes with the lectures, I feel like you can tell that like you know a lot about the subject, and I feel like your teaching will reflect that, and that like could you just like dumb it down a little, honestly? Like,
Yeah,
I agree.
For me, it's more
What'd you say,
like
Jordan?
it
What'd you say, Jordan?
moves fast, so I can't keep up. When it moves like really fast, I'm like, wait, I was looking at that slide and then you changed it, and I'm like, oh wait.
Okay, yeah,
redirect.
actually that's one bad habit I have, is if I'm in PowerPoint and I'm drawing in red and then I suddenly delete it and you guys are like, yeah, that's hard for me in my flow to do. So I'll try to recalibrate that again. I mean, it
That's
moves
what I am.
fa-
I went through an entire notebook already. Like I'm on my second notebook right now, just
I'm
because.
on my third.
Yeah, that's crazy. You know, you tell
Wow.
people this and they look at you like you're insane. Like literally, like my mom's like, "When's the last time you left the house?" And I'm just like,
Oh,
I don't know.
gee.
like all like so I like to write everything down. I like to write everything down so I can read it, and so I can like read it out loud to myself, like at like
Okay,
midnight.
question.
Like I do.
When you take your notes, do you write in full sentences?
Yeah.
Don't. That's the thing you can stop doing right now.
But
Write in sentence fragments. Because here's what's happening. The learning isn't happening when you go back and reread it. The learning is happening when you first write it.
Mhm.
Okay, let me show you something. So I told you I'm learning microbiology for the first time this semester. So one of the things that I'm having to do is like what you guys are doing is I'm taking notes. So ready here, this is me learning so there's the microbiology textbook. And then here are my notes. Okay, so I'm not writing in full sentences. I'm writing in fragments because what's happening is as I'm writing here, I'm building the patterns in my brain. And sometimes I'll go back and I'll just glance at this, or sometimes I'll rewrite it again. That's how the learning happened for me when I was in your seat. Was I would take my lecture notes, which were chicken scratch. Right after class, I would take my textbook and whatever handouts we had, and I would carefully rewrite it all and in a way that made sense to my brain. And then when I was going to go back to take to study for the test, I would take those rewritten lecture notes and write them again. Okay, the learning happened when I was writing it. Because when you're writing, you're a little slow, you're slowing down, right? You are putting them in your own words, you're thinking about it, and even better is we're talking out loud when you're saying it, right? So Tori, use those pieces of advice to maybe enhance your own studying like we were gonna talk about earlier today.
Yeah.
So I'd say outline the chapter in your own words, not full sentences. This protein is like that. You just wasted 3 seconds of your life, right? E2F equals positive regulator. The transcription factor E2F is a positive regulator. Right? So just like say it in the shortest way possible, because it's the key is what's the gold part of the information? What's the important part of the information?
Okay.
Okay. You also save notebooks.
Yeah, it's crazy the amount of paper I go through. I thought high
Yeah.
school was, I went through a lot of paper.
Is it okay if I email you later tonight? I
Yeah,
have,
I enjoy my next call.
Yeah.
yeah, I have to like go, but I still have a question.
This
So-
Zoom meeting has been recording the entire time we've been doing all this.
No.
Whatever. I'll stop recording.
Well,